Okay, so good afternoon. Can you hear me? Yes. Can you hear me? Okay, can you hear me? Okay, all right. So we are about to start our up to speed sessions, the fourth session. Okay, so very soon these sessions will be over. You can hear me, right? If I talk in this volume, you can hear me. Yes, I'm having a bit of a upper respiratory tract infection or something, probably viral or something. Okay, if I talk in this volume, you can hear me, right? Yeah, it's okay. Video, audio, all good. Neither? So we'll start in another 3 minutes. Okay. So by now you are used to the program, I hope. And so last week we finished everything up to microscopes. And from microscopes, we did this comparison between transmission and electron. Today, I'll show you some electron micrography images. I'll show you some of the electron micrography, and then today we have to just continue from microscopes onwards. I'm planning to finish most of the organelles. Okay, so because next week it's going to be cell cycle dedicated. Okay, cell cycle dedicated session, and then it's going to be, I'm going to give you a basic overview of enzymes as well, because I know most of you joined in the middle of enzymes somewhat. Results came actually before I started enzymes, but most of you guys started turning up at the classes in the middle of enzymes. Okay. Okay. නිකන් bio කරන්නම් කියලා එකපාරටම idea එකක් ආවා. Okay. So I'll be covering, giving you overview of enzymes as well. Because all of this is done, I'm going to cover everything because on 13th you are going to have a small paper. So before that, we'll be able to finish those sessions, and you will be able to go to face the small paper without any worries. Okay. So we have up to speed sessions on 3rd August as well as 10th August. So exact timing is perfect, so that you can go to the exam, go to the paper without a worry. So the paper will be given for the Thursday people on 13th and for the Saturday people on 15th. Okay, now listen. The paper will be delivered to you. Okay, if you guys didn't listen, Thursday people, if you are here at the meeting, Thursday people, online, okay, online, physical, of course, you can write at Headway Nikambo. If you have online people Thursday, if you didn't get the paper delivered, do not worry. Okay, that is because either there was a small delay. Okay, so if you did not get the paper delivered, you can write the paper by joining online at, so the paper delivery. So after you have received the delivery, you should join the, video. I prefer if you can switch on the video so that you can get a bit of exam, let's say, experience as well, with everyone switching on video so that I can see what you are doing as well. Okay, so what my point is, I won't be sharing the PDF on Thursday. Okay, if you didn't get the delivery of the paper by Thursday, you don't have to worry. Okay, you can join with the Saturday group. By then, you might have received the paper, or on Saturday, I'll be releasing the PDF. Okay, so because it's understandable if I release the PDF on Thursday, Saturday people will be able to see it. Okay, so also it's not that much of a paper. Hari the pair 15 MCQs, one structured essay and one essay. Hari? So it's nothing much. So immediately after you have done writing the paper, we are going to start the third lesson, evolution and diversity. For that, so there's a brand new tute, actually two tutes, and yeah. So I'm sort of finishing up that tute, and it will be soon delivered to you. Right now, I'm finishing up that tute. It will be more colorful than ever, and there's going to be a lot of let's say, proper biology is going to be there. This is mostly chemistry, no? Right? We are learning in the second lesson. So there's going to be some proper biology in the second lesson. Third lesson, evolution and diversity. Therefore, hope you are going to be all, let's say, hyped up from the third lesson at least onwards. Okay, because we are going to sort of who's doing that? Okay, right. We are going to actually, we are going to go with a bit of a increased pace from the third lesson onwards. Where properly we are going to start with going with an increased pace so that I can reach my syllabus coverage goals. Okay. Any questions before we start? Anyone having any doubts? Anyone who's not in groups? Anyone not in any of our groups? There's an announcement group, Headway, ISM. Everyone online should be in the announcement group. Okay, announcement group. That is an announcement community. I mean, the community is there, and in the community there are two small subgroups. But mainly, you should be in the community. That is where I put the announcements. Okay, so anyone who's not in groups, send a message via Zoom. My team will add you guys to the groups. And anything else? Tute delivery issues? Before we start. You didn't take the tute? Anything? I don't have a tute. Send me your number, my team will contact you. If you enrolled and enroll to this course, our Saturday or Thursday course, I think it should be on the way. By now, you should have received. Okay, I'll ask the website to contact you. There are hotline numbers in all the groups. Okay, there are two hotlines for my class. I mean, to handle any issue, and also there's a special number from the website that will help you anytime with any payment issues, any website issue. If you can't access recordings, if you can't upload payment slips, if you can't you can contact the website number. All those are I frequently send to groups. Okay, right. Okay. Anything else? Any other complaints? Anything else I have to solve? How's A-level life? How's physics and chemistry? Chemistry should be fun these days. Chemistry is, I mean, not that bad. Earlier chemistry is actually good. Derivation of this molecular structures, Vespa theory, those are really, I mean, there is some proper chemistry, so not that bad. Physics, well, what's happening? Mechanics started? Projectiles? Yes, good. Okay, right. Okay, everyone take out the resource book. Abhi, we'll get to biology. We get to biology, and so everyone has got the PDF. I have sent the PDF of the resource book everywhere. I have sent the PDF of the resource book everywhere. So you could have either bought one from the Ministry of Education, or you could have got a nice printout and got maybe a spiral bound with. I have seen a lot of versions. There's like a B5 size resource book as well. Maybe this iPad size again, right? B5 size resource books are there. I mean, that is also a good idea because it's kind of portable, so you can take it almost anywhere, and you can have it with you whenever you are traveling on the bus or going on a private trip or something. You can just take the resource book, okay, and do a bit of highlighting and do a bit of studying. Okay, so the portability of the resource book is also very important. Hurry. Okay, come to page number 24. Come to page number 24. So we'll have some more discussions, and I'll show you some of our new surgery videos. We'll talk about, and any other issues you can have, you can ask anytime. Okay, we'll get to the resource. So today we are going to start the actual cellular structure. Okay, we are going to start actual cellular structure. Now you can understand what order we are following in this lesson. So we started with actually the elements. Then we started how the elements or the atoms get together to form certain molecules. We went into a lot of depth about the major classes of molecules that are found in organisms. Then we talked about as humans, what are the instruments that we have to observe cells, because mostly at a cellular level, cells are very small. So in order for that, you require certain instruments that are known as microscopes. Main microscopes are developed for the study of cells. Can anyone tell me what is the study of cells known as? What is this term given to the study of cells? It is called cytology. So cytology වලින් එන what is the other next level of study after studying cells? What is the next level of organization? Order and organization, what is the next level after cell? What is the next level after cell? Yeah, cells form tissues. Ne? Tissues, what is the study of tissues known as? Histology. Histology, yes. So histology. Okay. So tissues get together to form organs. Organs form organ systems. Organ system. So an organ system, several organ systems get together to form organism. So actually, this rough anatomy from everything above. So I just told you, right? So everything usually above the tissue level, we usually study under the what science, sort of what branch of biology. We usually call it the study of anatomy. Right? So in your first year in medical school, if you are doing sometimes in certain science degrees also, you have the science, the subject of anatomy, where you do the dissections and other things. Right? For there, actually, in our when we were studying anatomy, we had actually histology as a let's say under the subject of anatomy. We had histology. We had microscope and histology lab sessions. So under the science of anatomy itself. Hari. So as I told you, now we are sort of relating. Can you see from the atomic level? Now we are coming into the actual cellular level. Okay. So we are going to learn about in detail about the interior of cells, the organization of cells, different structures found inside cells, and how all those cellular structures get together to make the cell the simplest functional unit that we can call as living. The cell is the smallest thing that we can call as living. A single cell is the smallest thing that we can call as living. Understood? Right. So we'll read about it as usual. Okay, I want you to highlight the resource. I want you to highlight the resource as you are reading. And so the resource book customization. I call this highlighting and writing small things on the resource. It's customization. You have to customize it. That is one of the main things about A-level biology is how you handle your resource book. Resource book customization is very, very important. Correct? So keep it neat. Do not make it a mess, but keep it very neat and also very visible so that you are you actually like to come back to the resource and actually study it. Got it? Let's read. Cell theory. Okay, all organisms are composed of cells. Recall the hierarchy of life. Okay, the basic unit which can be called living is the cell, which may form a single organism such as chlamydomonas or yeast, or a multicellular plant or animal. Okay, you know chlamydomonas. The chlamydomonas is what we call as a protozoa. Chlamydomonas, you can remember it. It's sort of a bit of a photosynthetic. This is a photosynthetic organism we have in your syllabus. Okay, that is a protozoa. And yeast, you know yeast, small single cells of what do you use yeast? There are several uses. What is the main earliest use of yeast? What do you think? Historically, what is the earliest use of yeast? Yeah. With the human population, what is the earliest use of yeast that we used? What do you use yeast for now? What do you use yeast right now, Monado? To make bread. Because when you put yeast into a mixture of mixture containing sugar, maybe yeast cells now, this yeast is going to produce by its respiration. That's what we have started in our regular theory class. Sorry, when they start to produce carbon dioxide gas. So when the carbon dioxide gas is released, this bread dough is going to have this small the crumb texture. Okay, so that the bread dough rises up again pippen out here, right? So that is not actually the first use. Right? So yes, you need yeast to make bread. Right? That is not the human civilization's yes first use. Right? Historically they have found out yeast was first used to make alcohol. Okay. So similarly, when you have a sugar source sugar source කියන්නේ a sugar containing substance. For example, you make alcohol in Sri Lanka using coconut sap, coconut phloem again. Then sugar molasses you can use. That is after extracting sugar from the sugar cane, you can use the remaining solution, right? Whatever, make a yeast add karahama, you are going to get. He's going to produce ethanol. He's going to produce ethanol. Right? Before people started to make bread from yeast, early people started to make alcohol from yeast. Okay, so that's there you can see where the priorities are for the human population. Right? කන්න නැතත් බොන්න තියෙන්න ඕනේ. Okay, right? No, that's actually the historically first use because they found out in these certain plant saps, right, or phloem saps වල naturally තියෙන phloem saps වල there is yeast floating in the air. So in those plant saps, naturally yeast grows, and someone who carry adventurous must have drunk that fermented plant sap and gotten drunk, and they all started to enjoy it, right? Before started making bread. Okay, right. Can you highlight the next sentence in the resource? Come to the resource. The cell is the basic structural and functional unit of life. The cell is the basic structural and functional unit of life. Okay, the cell is the basic structural and functional unit of life. Okay. The level of organization of matter represented by a cell shows all the characteristics of life. Any stage below this level of a cell cannot be considered living, whether it's a single cell organism or a multicellular plant or animal. Right. Then Robert Hooke, highlight Robert Hooke name, examined a cork using a simple microscope and gave the term cell. Highlight Robert Hooke and gave the term cell. Main Robert Hooke, he actually had a very basic microscope. Okay, this is actually cork under his microscope that he observed, right? So Robert Hooke was one of the pioneers of study of cells. Actually, he gave the term cell. So he started this. Got it? Methane ලියාගන්න. So we have to write some things in this space. We have to write some things in this space. So don't come to this side. Methane ලියාගන්න. Robert Hooke, under this diagram, write down. Robert Hooke gave the term cell, which is a very important milestone. It's a very important milestone. Robert Hooke gave the term cell. Okay. Then Anton van Leeuwenhoek, a contemporary of Robert Hooke, was the first to describe and record living single-celled organisms such as euglena and bacteria. So he observed some of these pond water sample, and he observed certain moving small organisms there. He actually discovered, he didn't know what they were, but he described them. Okay, these are very small scale microscopic organisms. So Anton van Leeuwenhoek, can you highlight Anton van Leeuwenhoek? Was the first to describe and record single cell organisms, euglena and bacteria. Okay, euglena and bacteria. Euglena and bacteria. Matthias Schleiden. So you have the three people next. Hari. So we start with Matthias Schleiden. He's a botanist. Can you highlight it maybe in sort of underline in greenish pen or something, Hari? Matthias Schleiden was a botanist. Botany is what? Study of plants. He studying plant tissues concluded that all plants are made up of cells. Can you highlight it? Matthias Schleiden. He concluded that all plants are made up of cells. Then next, Theodore Schwann, he was a zoologist. Zoologist is study of animals. He's concluded that animal cells are also made up of cells. So we have two people now. We need three. Okay, we need three. Then Rudolf Virchow showed that all cells arise from pre-existing cells by cell division. Can you highlight? They can ask you, what did Matthias Schleiden say? What did Rudolf Virchow say? What did Theodore Schwann say? They can ask in the structure essay paper. Right? Rudolf Virchow showed that all cells arise from pre-existing cells by cell division. which is an important concept. Right? Cell division also important concept. Uh, so all three of these people, Schleiden, Schwann, and Virchow, got together to present what we consider in modern times as our modern cell theory, right? So, මෙතනින් අපි ලියාගමු, right? We'll write down here cell theory was by, you only need to know actually the three main names, Schleiden, Schwann, SCH, no? Schwann and Virchow. They presented the cell theory. I want you to write down the three components. May these three components, Hari, listen. These three components are the fundamentals of biology. These are fundamentals. I told you there are certain core rules, or let's say core principles or core knowledge that you need to have in biology. Similarly, for to the chemistry, we got the atomic numbers, molecular mass. So again, these are core knowledge, right? So similarly, in biology, you have some fundamental principles or knowledge that you need to have. You can't say you are a biology student, right? So, such an important place is this. The cell theory, the three postulates under the cell theory. Alright? Let's read in the resource book. All organisms are composed of one or more cells. Either organism is a unicellular or he has more than one cell, which we can consider as multicellular. Second one, basic structural and functional unit of organism is the cell. Okay. Then third one, all cells arise from pre-existing cells. So you might ask, okay, what cell did the first cell arise from? That is what we are going to learn in third lesson. Where we are going to see how the first cells were produced. That is, there's a nice theory, biochemical evolution. Big Bang again, we are going to come to the origin of humans in a Kelima story like way, which is very interesting. Okay, but anyway, these are the three postulates of cell theory. Please write down here. All organisms composed of one or more cells. If you have a single cell, you are unicellular. If you have more than one cell, you are multicellular. Basic structure and functional unit of life. Basic structure and functional unit of organism. So you can say organisms is the cell. These three things you have to have in your memory almost exactly word by word. Hari, all cells arise from pre-existing cells. All cells arise from pre-existing cells. Okay, quickly take this down. Okay, right. Okay, then we can talk about the next topic. Okay, come to page number 25, organization of cells. There are two kinds of cellular organization, prokaryotic and eukaryotic. Highlight that I have two kinds of cellular organization. I'll show you with the software, right? What is the prokaryotic, classical prokaryotic, what a typical prokaryotic cell looks like, what a typical eukaryotic cell look like. Okay. Then all cells share certain basic features. All cells share certain basic features. Those are also so important. Right? These four, they are really important. Okay, let's see. All cells are bounded by a plasma membrane, which is a selective barrier. Now listen, if there's a cell on Earth if there's a living cell on Earth, that cell should have a plasma membrane. Plasma membrane, which is our actually the next topic that we are going to learn. We are going to draw a nice big plasma membrane here. Okay. Within the cell, there's a semi-fluid jelly-like substance which is called the cytosol. Subcellular components are suspended within the cytosol. Can you highlight? Semi-fluid jelly-like substance called cytosol. Then highlight subcellular components are suspended in the cytosol. Subcellular components are suspended in the cytosol. Third one, carry DNA as genetic material. If you are again a living cell on Earth, you are going to have DNA as your source of information. All your information is going to be stored in DNA. Correct? Nothing else. There are some viruses that have RNA as their genetic material, but viruses are not considered as living organisms. Viruses are not considered as living organisms. You will understand why. They only show. Can anyone tell me if you take a virus, what is the only characteristic of living shown by a virus? So now you need to know what are the characteristics of living. You need to know what are the characteristics of living. You have order and organization, metabolism, irritability and coordination. Yes, evolution, reproduction. Yes, yes. Actually, it's not actually a reproduction, it's actually a replication. It is going to try to copy itself somehow. A virus is going to try to copy itself. For example, what I'm having right now probably is a virus. The virus entered my upper respiratory tract and it has entered into my sort of the epithelia, I can inside the nose. I'll show you if I have some time. Inside the nose, there's a lot of tissue that are there to sort of warm up the air you breathe. The air you breathe should be warmed up. Otherwise, too cold air could damage your lower respiratory lungs. Right? So your nose is filled with a lot of tissues. So once a virus gets in there, the virus can enter into those cells and it multiplies in those cells by damaging the cells. Okay. So when it is multiplying, the cells are damaged. So because the virus is going to sort of hack into our body cells, and it's going to use all of our cellular resources, all the enzymes, all the proteins available inside the cell, just to make more copies of the virus. Virus take copies then it's going to use all the cellular resources. So that's why viral diseases damage our body cells. Okay. So viruses, they are only focused on mainly multiplication, or you can say reproduction. That is also only when, yes, they are inside a host cell. A host cell, so now could be my upper respiratory cells inside my nose, or our upper pharynx area, throat area. So they are inactive when they are in the environment, they are floating in the air. They could be on certain surfaces, on your hands, on things you touch, okay? Anyway, finally, ribosomes are found in all cells. Ribosomes, will study the structure of ribosomes. Ribosomes are found in all living cells because they are essential for the protein synthesis. They are essential for protein synthesis. Right? Okay. Let's move on. So Then on page number 25, there's a chart actually. There's a comparison between prokaryote and eukaryote. In my normal theory class also, I am going to skip this for now. Okay, I'm going to skip this chart for now because we are going to come back to this chart after we have studied almost all the organelles. Then we will understand it much clearer because there I don't need to talk about genetic material being circular, those things, and also the structure of cell walls and the structure of flagella. Those things will be easy. I mean, it will be much easier for you to understand once we have covered the rest of the organelles and cellular structures. We'll come back there. Right? Okay. So for now, let's skip those two charts, and can you come to page number 27? We are going to go into a big topic, that is our plasma membrane. Very important. Very important. Essay value as well as a lot of MCQs have been asked from the plasma membrane. Okay. Now listen. What do you mean by the plasma membrane? Plasma membrane. Any living cell, I told you, they have to have a boundary. No? The cell has to have a boundary. That boundary in all living cells is composed, I mean, is going to have a similar structure. That is the structure that we are going to study today. So if you take a typical animal cell like this, Sarthak, if you take a typical animal cell like this, you can see maybe this the outermost structure here. This whole thing is what we call the plasma membrane. So plasma membrane is actually very thin. It is only two phospholipids thin, right? It's only two phospholipids thick. So it's a very thin structure. It is the outer boundary of all living cells. So if you take, so this is a normal animal cell. No? So if you take a plant cell outer to the plasma membrane, you are going to have a cell wall, ne? A cell wall composed of cellulose mostly. You are going to have a cell wall. So even though they have a cell wall, inner to the cell wall, they are going to have a plasma membrane in a plant cell as well. If you come to a prokaryotic cell, so this is what we call as a prokaryotic cell. This is a typical bacteria. Bacteria can have a outer what we call as a capsule. So having a capsule makes a bacteria difficult to destroy. Antibiotics can't go and destroy the bacteria if they have this protective covering. But even though inside the bacteria can have a cell wall as well, here even in prokaryotic cells, you can have a plasma membrane. Got it? So the structure of the plasma membrane is what we are going to learn now. Yeah. Any questions? Hey, come back to the resource again. Plasma membrane. Let's read. Plasma membrane is the outer limit of the cytoplasm. All cellular membranes resemble the ultrastructure of plasma membrane. That is important. Okay, if you have a membrane inside your cell, that membrane structure is going to be equal to the structure of the plasma membrane. For example, we are going to learn about plasmodesma. We are going to learn about endoplasmic reticulum. We are going to learn about the membranes of chloroplast and mitochondria. All those membranes that are found inside the cell also, they are going to have the same organization, or let's say structure, as the plasma membrane. So plasma membrane is the outer covering, yes, but if you have a membrane inside the cell, it's going to have a similar structure to the plasma membrane as well. That's what we say. All cellular membranes. So cellular membranes can be inside the cell or outside the cell. No? Okay. In 1972, Singer and Nicholson put forward the fluid mosaic model of cell membrane. Highlight. Okay. 1972, Singer and Nicholson. What did they put forward? Yes. Fluid mosaic. Fluid mosaic. Okay, fluid mosaic model. Mainly composed of phospholipids, the most abundant type of lipid in plasma membrane. Highlight phospholipid. and then protein. Hurry. Now listen, we are going to draw our plasma membrane, which is one of the most important diagrams that you need to be able to draw. Okay, that is the structure of the plasma membrane. So now listen, you need, I think we have drawn together the structure of phospholipids. How do you describe the structure of a phospholipid? A phospholipid has a hydrokernel. What are the two parts of a phospholipid, first of all? A phospholipid has a head as well as two tails. A phospholipid has a head and two tails. So the heads and the tails have two different properties. The heads and the tails have two different properties. What is the property of the head in relation to water? I'm talking about in relation to water. What is the property of the head mainly in relation to water? The head likes water. The head likes water, so we call it as hydrophilic head. Okay, it likes water. Hydrophilic. So this is the head. These are the heads. These are the heads. And these are the tails. So what do you call the tails? What is the nature of the tails? They don't like water. So if you don't like, or actually we call them, they are afraid of water. So they are hydrophobic. They are afraid of water. So your plasma membrane is, first of all, is a phospholipid bilayer. Bilayer means what? It has two layers of phospholipids arranged like this. It has two layers of phospholipids arranged like this. So now can you understand why are we drawing the hydrophilic heads? Why are we drawing the hydrophilic heads facing outwards? Why are we drawing the hydrophilic heads facing outwards? That is because this is the inside of the cell. I'm drawing with the blue color, right? This is the inside of the cell. Okay, you can say intracellular side. This is the inside of the cell. You can say intracellular side. Right? Then this is the outside of the cell. So we can call it the extracellular side. This is the outside of the cell. Okay, extracellular side. So I'll actually bring these a bit more lower, right? That is the extracellular side. So what do you have usually? Imagine in the cytosol, that is the inside of the cell, the jelly-like material you have inside the cell. What do you have most? What do you think you have most? It's actually water, right? On the intracellular side, what you have most is water. Also, if you take a multicellular, like imagine our muscle tissue, right? If you are a muscle cell, what do you have usually outside? You are mainly something you have something called tissue fluid that is mainly coming out of the blood, no? So in usually living cells, there is water inside as well as water outside. So the hydrophilic heads, that means the heads that like water, they are going to be facing inwards and outwards. So this is the intracellular side. We can call this is the extracellular side or outside of the cell. This is how the phospholipids arrange themselves. Why? The tails don't like water, so they have to be protected from the water. They can't live with water. They have to be protected from the water. Therefore, the heads are going to face into the water and the tails are going to be to the inside. Then we can say, what else did we say? The phospholipid bilayer is, the plasma membrane is composed of phospholipids and proteins. Now let's draw some proteins. There are so many different types of proteins. Okay, there are so many different types of proteins in the plasma membrane. Okay, that they have such a variety. And it's important that you understand. Okay, so we'll draw one of the types of proteins that we have in the plasma membrane. Now you can see this is a big protein. This big protein extends the entire thickness. No, so this is the thickness of the plasma membrane. No? So it sort of it goes across the entire plasma membrane. So this type of proteins we consider as a transmembrane protein. We call this as a transmembrane protein. Usually transmembrane proteins act as what we call as here, channel proteins. Okay, they usually act as channel proteins, that they have a this sort of an inner channel that through which different ions and other things can pass. Okay, they have an inner channel through which ions and other things can pass. Okay, so this is what we call as a channel protein. It is also a transmembrane protein. This is a channel protein as well as a channel protein through which several things, many things can pass through that. Alright, so we are going to continue the plasma membrane again. We are going to continue the plasma membrane. That is by drawing some of phospholipids. Then I can draw another type of protein. That protein, now listen, that protein doesn't go the entire thickness. That protein doesn't extend the entire thickness. So it is only extending only maybe this much. This much. Okay. So we have a special name for him. I'll tell you, right? So he's not a transmembrane either. What you can say, what he's not, if you have to be a transmembrane protein, you have to extend the entire thickness. So if you are only extending this much, you are not a transmembrane protein. Okay, you are not a transmembrane protein. Right? Keep that in mind. So we'll draw some more phospholipids. We'll draw some more phospholipids. Okay, and more phospholipids. We'll draw another one of these small proteins that are not exactly transmembrane. We can use these proteins can have so many different compositions. Okay, so this is such a protein that is not a transmembrane protein, because why? It's not a transmembrane protein. It is not extending the entire thickness. No? So this is actually the classical composition of your plasma membrane. The classical composition of your plasma membrane is a phospholipid bilayer with proteins embedded. Okay, it's a phospholipid bilayer with proteins embedded. Then there are some other type of protein, now listen, that are not actually inside the plasma membrane at all. They are not in the plasma membrane at all, but they are just loosely bound. We say they are loosely bound to the inner, or it could be the outer, inner, or it could be the outer side of the plasma membrane. So I'll draw one here. Okay, they are just loosely bound to the inner or the outer surface of the plasma membrane as well. So all these are proteins, all these are proteins, and the rest is phospholipids. That's all we drew. Now come back to the resource and let's read what we have drawn so far. Plasma membrane has the following features. It is about 7 nanometers thick. Highlight. It is about 7 nanometers thick. Okay. Seven nanometers. So that's actually a, let's say a common thickness, because I told you in all living cells you have the plasma membrane. That plasma membrane is going to have this same structure. Right? Mainly made up of a phospholipid bilayer. Why do you call it as a bilayer? It has two layers of phospholipids. Simple. Right? Phospholipids are amphipathic molecules. Highlight. Phospholipids are amphipathic molecules. Why do you mean by amphipathic? Let's see. Hydrophilic heads of the phospholipids face towards the aqueous environment of both inside and outside the cell. Right? Amphipathic means, listen, if you have both hydrophilic and hydrophobic natures in the same molecule, we call it as amphipathic. Okay? Amphipathic. You can write down if you like. Okay? Actually, you don't need to. Okay? Amphipathic. If you really want, you can write down. Having both hydrophilic and hydrophobic nature, in the same molecule. Amphipathic means having both hydrophilic, that means water loving, and hydrophobic, nature on the same molecule is known as amphipathic. Is known as amphipathic. Right? Both water loving and water fearing, hydrophobic and hydrophilic natures. Keep reading. The hydrophobic hydrocarbon tails face inwards and creates a hydrophobic interior. So this is what we call as the hydrophobic interior. Maybe the center of the plasma membrane is having a hydrophobic interior. So what did we learn right until now? These two phospholipid layers form a bilayer. So you call it as phospholipid bilayer. This is known as the phospholipid bilayer. Plasma membrane is compared to the fluid mosaic model. Since phospholipid molecules are movable, they provide the fluid nature to the membrane. Right? So these phospholipids now, they are not locked into place. Then if you take a brick, all the bricks are immovable. Right? Gadol gattut, gadol bittiyak, they are immovable. Right? It's not like this. This plasma membrane is like if you take like a set of plastic balls floating on top of a swimming pool, right? So they can move with way shake. I mean, when the water is moving, they can move, right? So this plasma membrane or the phospholipids are also like that. They can move. They are fluid. So phospholipids contribute to what nature of the plasma membrane? Fluid nature. Fluid means sort of be able to change. Got it? Since phospholipid molecules are movable, they provide the fluid nature. Can you highlight? Phospholipids provide the fluid nature. Phospholipids provide the fluid nature. Hari? Phospholipids provide the fluid nature. Kattiya not sleepy neda? We just started, no? Demmali dayan පටන් ගන්න එපා. තව පැයකින් විතර නිදා ගන්න, okay? Hari? Then, protein molecules embedded randomly contribute to its mosaic nature. Highlight. Protein molecules embedded randomly contribute to the mosaic nature. What do you mean by a mosaic? Right? If you go to a church, even in sort of Buddhist temples, on the floors, and in church, certain sort of these old-fashioned churches, I don't know if they have in mosques or something, there are these sort of different patterns or certain diagrams that they have made using piece of tile, colored tile, or it could be on the floor, right? Could be on the walls, right? Made up of tile pieces, color tile pieces. Have you seen mosaics? Okay, that sort of a nature is there when you take a look at this plasma membrane from the top. I think in our tube there's a diagram of our fluid mosaic model. I think in the previous tube, didn't we have? I can't remember where exactly I put it. I can't exactly remember. Was it in our previous tube? Did we have a plasma membrane 3D diagram? Where was it? Did we have it in the before this? Our first tute? I think there was one. Amity? Yeah, this one. So can you see? If you take a look at the top of this is the plasma membrane, right? Yeah. If you take a look on the top of this plasma membrane, you will be able to see it is a sea of hydrophilic heads. මේ බෝල බෝල වගේ තියෙන hydrophilic heads. And there you have randomly proteins here and there. Okay? So if you pull these two layers apart, if you take apart the phospholipid bilayer, you can see the interior as well. Okay? This is actually a nice diagram of a more detailed diagram of plasma membrane. We are drawing a much more simple one. So you can see there are so many different types of proteins that you need to know. Okay, so we'll draw the diagram first, and then we'll come back to this one again so that I can show you. Where is it? Okay, make it. Okay, right. All right. So keep reading. So proteins molecules embedded randomly contribute to the mosaic nature. Did you highlight? Proteins embedded randomly contribute to mosaic nature. These are called integral proteins. If you have any protein that is embedded, embedded we can know that they are inside the membrane somehow. Could be halfway, or could be they are go completely through the entire thickness. Doesn't matter. They are known as what? Integral proteins, right? So now listen. These are math integral, math integral. All these three are integral proteins. Some of the integral protein molecules penetrate all the way through the membrane. These are called transmembrane proteins. Alright. So now listen. Out of the integral proteins, a special type of protein is there. That is the proteins, that is the integral proteins that penetrate the entire thickness. They are known as by a special name called transmembrane. Okay. So this is an integral protein, but it is also a transmembrane protein. It is an integral protein as well as a transmembrane protein. Can you understand? This one, this red color one, because why it is goes the entire thickness, no? Most of the integral proteins are transmembrane proteins which have a hydrophilic channels. These act as pores through which certain ions and polar molecules can pass. So you can label this channel that is going through this, you can call it as a hydrophilic channel. It's a hydrophilic channel so that certain water soluble substances can pass through the transmembrane protein using it as like a door or like a pathway, gateway. Okay. Keep reading. We are in the next page. Page number 28 of the resource book. Okay. Some proteins are not embedded in the bilayer at all and are loosely bound to the surface. They are known as peripheral proteins. Mainly. Now this is a peripheral protein. They are not in the plasma membrane, but they are loosely bound. Okay, so it's a peripheral protein. This is also a peripheral protein. This is also a peripheral protein. Okay. Some proteins and lipids have short branching carbohydrate chains like antenna. Okay, before we move on to that, I want you to, there's a small classification that I want you to have. We'll write it here. Don't start writing yet, otherwise we'll run out of space. Okay, so we can categorize integral proteins. You can have integral, sorry. You can categorize plasma membrane proteins as two types. You can categorize plasma membrane proteins as you can categorize plasma membrane proteins as proteins that are embedded in the membrane itself. Proteins that are embedded in the membrane itself, they are known as integral. They are known as integral proteins. Then there are proteins that are not embedded in the membrane at all, but they are loosely bound. So they are called as peripheral. They are known as peripheral proteins. Out of the transmembrane proteins, you have a special category of out of the integral proteins, you have a special type of integral protein that is known as transmembrane membrane protein. And out of the integral proteins, you have a special type of uh, integral protein that is known as transmembrane protein. So you can say also plasma membrane has a fluid nature as well as a mosaic nature. Fluid nature is by who? Kiana. Fluid nature is given by what? By phospholipids. Mosaic by proteins, not just any proteins. You have to say embedded randomly. The proteins are embedded randomly. Then those proteins are the plasma membrane proteins. Plasma membrane proteins are of two types, as integral proteins and peripheral proteins. Integral proteins have a special subtype as the transmembrane proteins. Correct? Up to now, all clear? Okay. Right, moving on. Come back to the resource. Some proteins and lipids have short branching carbohydrate chains like antenna, forming glycolipids and glycoproteins respectively. Alright, now listen. If you take a protein like this, if you consider a protein like this, I'll erase this peripheral story, okay? I'll erase this peripheral, right? Because I need to draw a protein there. Okay, if you take a protein such as this, it can have branching chains of carbohydrates. It can have this sort of branching chains of carbohydrates that we call as what? So they are carbohydrates, so they should be glyco. They should be glyco. So since they are attached to a protein, what can you call them? You can just call them as You can call them as a glycoprotein. They are known as glycoprotein because they are connected to a protein. Then also what you can do is you can have similar carbohydrate chains connected to a phospholipid as well. You can have a similar carbohydrate chain connected to a phospholipid as well. So actually I'm running out of space, sorry. So you can make it more neater, right? So this sort of a carbohydrate chain arising from a phospholipid. So it's a phospholipid, no? So since it's a phospholipid arising out of a lipid, it's a carbohydrate chain arising out of a lipid. So you can call it as glycolipid, ne? You can call it as a glycolipid. Understood about glycolipids and glycoproteins. So if there is a carbohydrate antenna-like structure coming out of a protein, it's a glycoprotein. Right? If it's coming out of a lipid, it's a glycolipid. Right? Okay. Then what else do they say? Okay. Some proteins and lipids have short branching carbohydrate chains like antenna, forming glycolipids and glycoproteins respectively. Right, keep reading, very important. Next, animal cell membrane contain a few cholesterol molecules randomly integrated into the lipid bilayer. Okay, these cholesterol molecules provide rigidity and stability. Highlight, animal cell membrane can have a few cholesterol molecules. They provide rigidity and stability. Two sides of the membrane may differ in composition and function. Okay, now this cholesterol, there is a few things to be explained. Okay, cholesterol, now listen. These cholesterol molecules can be found in between these certain phospholipid molecules, only in animal cells, Certain cholesterols are only found in animal cells. Cholesterol molecules, they are there to provide, particularly in certain temperature changes, the cholesterol molecules can maintain the stability. So when the temperature gets too high, these phospholipids become actually too mobile. These phospholipids become too, let's say, too mobile, so that they will start to move too much. They will start to move too much. Therefore, what needs to be done is, in order to maintain the stability of this plasma membrane, you need the help of a molecule. You need the help of a molecule such as the cholesterol. Okay, you need the help of a molecule such as a cholesterol. Okay, right, to maintain the nice stability of this membrane. When the opposite happens, when the membrane becomes too cold, okay, they will start to freeze together. This membrane phospholipids will start to get locked into a crystal shape. Okay, so when that also happens, these cholesterol molecules try to keep it more loose, more fluid, right? So this is actually the phosphomembrane that you need to be able to draw. The labeling is very important. Okay, so this is cholesterol. The labeling is so important. Okay, so that you don't lose any marks. Okay, and also another thing is, be very careful when you are labeling which side. Intracellular side, you can also call it as the cytoplasmic side, intracellular or cytoplasmic side. This is the extracellular side. Okay, now you can start drawing this in page number 2. Okay, you can start drawing this. Phospholipid bilayer is this, label the extracellular intracellular side. This is the transmembrane protein, which are all part of the integral proteins. So this is also an integral protein, because why? They are embedded in the plasma membrane somehow. And this is a peripheral protein. There are special types of integral proteins that are known as transmembrane proteins. Transmembrane proteins have a hydrophilic channel, so you can do that labeling. And then connected to certain proteins, you can have antenna-like carbohydrates that are known as glycoproteins. Then connected to lipids, you can have such carbohydrate chains that are known as glycolipids. And then I want you to categorize, what are the two parts of the plasma membrane? There's a fluid part, there's a mosaic part. The fluid nature is given by the phospholipids, mosaic by the proteins embedded randomly. You have to say that. They can ask you in the structured essay paper, what gives the mosaic nature? Okay. Then, so out of the proteins, you can categorize the proteins as integral proteins and peripheral proteins. The integral proteins have a special type as transmembrane proteins. Okay, quickly take this down. Why is it called a bilayer of phospholipid? You can see, because there are two layers of phospholipids. No? This is one phospholipid molecule, this is the other molecule. So altogether, there are two layers of phospholipids. Okay. Nothing else to be written. In page number 2, there is nothing else to be written. Okay. All the things that have to be written here is I have written. Okay. If you can't read anything, let me know. No need to leave any more space. Right? Everything to be written, I have written. Okay. Be careful when you are writing the method. So I have labeled this as glycoprotein, and the glycolipid labeling is here. Okay. Uh, because we have to go fast, I prefer if you take a screenshot or something. Okay, then do it neatly when you have time, because I'm going to move ahead a bit fast. Okay, you can take a screenshot when I'm not here. You can start drawing a rough sketch. So in this space, you only need to draw this, okay? That's why I'm drawing in the same tute. I'm putting myself in your shoes, right? So I'm trying to do it in the space that you are supposed to do it, okay? So you can understand how the tute is going to be filled. And also there was a small discussion that I'm doing it completely in English. I'm teaching my class completely in English. Is that an issue for you guys? No, right? I haven't. Can you please upload the tute? What do you mean? Did you get the delivery of this tute? At this point, we have been doing this tute for a long time now, in the up to speed sessions and everywhere. Didn't you get delivery of this tute? Send me your number so that my team will contact you. Anyone who doesn't have the suit, send me your number so that the team will contact you. Because otherwise, it's at this point, if you haven't got the tube, that's a problem. I need to look into it. Okay. Send me your numbers. Send me your numbers. Online, right? You guys are online students, right? Otherwise, physical people, if I will definitely have handed you out. Anyone else? Taggala me kandina kama issues. Also, if the website tries to contact you, please answer. Okay. I have had instances where students are not answering phones because they say they haven't got the tute in the class. But they don't answer when they are contacted by the website technical people. Okay. They're just trying to help you. Okay. The tute is in the Zoom chat also. Hurry. It will be delivered soon once you enroll to the website. If you are physical, then I'll give it to you physically. Harita, this Thursday you can you can ask from me, or this Saturday. If you are online, will be delivered as soon as you enroll to the website. Hurry. Okay. Right. Did you do this categorization? Plasma membrane is fluid and mosaic two types. And then the fluid nature is by phospholipids. The mosaic is by the proteins embedded randomly. Okay, right. Moving on. We are going to move on to the individual functions of different proteins that we have. Right. Now listen. This is what I told you about the cholesterol. Okay. Now ignore for the top part for now. Cholesterol molecules that are available in the animal cell is going to perform this role. Right. Can you see it reduces Mikame? This is something extra. Reduces fluidity at moderate temperatures. Okay. But at low temperatures, it hinders solidification. Okay, at moderate temperature means at our normal body temperature, the cholesterols reduce the fluidity. What do you mean by reduce fluidity? That means it doesn't allow it to move too much. Very move anything. But at very low temperatures, it prevents solidification. Ice it doesn't allow the plasma membrane to freeze. Okay, so because you can understand they act as sort of let's say they sort of get in between phospholipids and prevent them from solidifying. Understood? There is a role of cholesterol in animal cells. Got it? Now we are going to move on to functions of the plasma membrane, which is a very important topic. Let's read functions of the plasma membrane. So what we are going to do actually is, in front of each of these diagrams, we are going to write down the functions so that you have a visual idea, right? With a diagram, you can remember the individual function. Okay. Right. So let's read uh, page number 28. Functions. Hi, let's read. Plasma membrane surrounds the cytoplasm of living cell, physically separating the intracellular components from the extracellular environment. Highlight. Surrounds the cytoplasm of living cell. Surrounds the cytoplasm of living cell. Ah, yeah, I couldn't ask. So no one has an issue with me talking only in English, right? After all, it is English medium biology, right? So you're going to be, I mean, it's important that you get familiar with the, let's say, language as well, because particularly in the essay papers, you have to be able to handle a little bit of English as well. So, Metike, watch some English movies, some good English TV shows, not very long ones. Some brain stimulating ones as well, some documentaries, historical ones. There are mini series, nice mini series. So watch those, Metike. Not anything is better than just scrolling through Instagram reels, TikTok reels, and YouTube shorts. They are going to ruin your attention span. Anything. Watch some good classical movies, good some documentaries. Okay. I can recommend, I mean, watch this one called Sherlock. Okay. Sherlock Balladin, everyone. And watch some of the Christopher Nolan's movies. Maybe uh, everyone should have watched this Interstellar and Inception. Those kind of brain stimulating movies you should watch. How to watch mean you can download illegally. Okay, there are online sites. I mean, you can watch the movie completely online, or you can just download. What website I use? Okay, there are torrent sites. No, you guys know this. Ask from your friends. Torrent sites. Yes, I don't recommend. I'm not going to be uploading this to you. Yeah, so don't say I ask you to torrent. It is illegal, but Omari Balan. What's your favorite movie? That's like you can't choose one favorite movie either. English newspapers. That's very old-fashioned, either. You guys read old newspapers. That's very old-fashioned. No, that is more entertaining to watch some good movies. Yeah? It's more entertaining to watch some good movies and TV shows. Yes. Then the news, just be stay updated of what is happening, and the news is pretty depressing. Ne? Nothing to gain from watching the news. Just stay in touch with whatever that is happening, just to have a rough idea. But news is very depressing. No? It's all politics and other things. So just focus on studying and maybe watching some new, so not new, some good movies. Anyway, highlight. Plasma membrane surrounds the cytoplasm of living cell. Highlight that. Second one, plasma membrane is selectively permeable and able to regulate the exchange of material needed for survival. Highlight selectively permeable. These are keywords. These are words that you cannot miss. So we'll read the properties, sorry, the functions list first, and then we'll fill this tube. Okay, right. Plasma membrane is selectively permeable. Now, what do you mean by selectively permeable? That means it acts as like a gateway, but it's a filter. No? So, for example, in the highway entrance, you have a toll gate. No? So, there you are only allowed, I mean, only the vehicles that are allowed to enter the highway is sort of filtered out. Ne? So, you three-wheelers, motorbikes, and tractors, or those things you can't enter the highway. So, the plasma membrane is like that. There are proteins that allow or regulate what are the things that are allowed to enter into the cell. Okay. Then third one, proteins embedded in the plasma membrane identify the cell, enabling nearby cells to communicate with each other, involved in cell recognition. Highlight cell recognition. Okay, cell recognition, very, very important. Right. So I think, okay, we'll finish reading everything. Some protein molecules act as receptor molecules for interacting with specific biochemicals such as hormones, neurotransmitters, and immune proteins. Highlight act as receptor molecules. We are only going to highlight the important thing. We are going to highlight the entire resource now. So highlight only the important words or phrases. Act as receptor molecules, then highlight the molecules, hormones, neurotransmitters, and immune proteins. Okay, I'll teach you everything later. Okay, next one. Some proteins in the cell membrane attach to cytoskeletal fibers and help to maintain the shape of the cell. Highlight attach to cytoskeletal fibers. Attach to cytoskeletal fibers and maintain shape of cell. Words stick with that. Highlight the important phrases. Right? Finally, some proteins act as enzymes. Highlight the enzymes. Right. So now we are going to do is, if you send to the main chat, I won't miss your messages. When you send me personally, I'll miss them sometimes. Okay. Okay. So what we are going to do is we are going to in front of each of these diagrams, we are going to write down the function that is indicated by them. Right. So this is actually the plasma membrane acting as a selectively permeable membrane. This diagram indicates the plasma membrane acting as a selectively permeable membrane. Here you can actually see the plasma membrane acting as a transporter in two ways, right? So you can put a label to these. These two are transport proteins, okay? So the transport proteins are actually of two types. Now listen. Transport proteins are actually of two types. You can have what we call as active transporters. Then active transporters is this. This is an active transporter. This one is an active transporter. You can see you need an energy payment. You need to make a payment so that this molecule can go across this transporter. Right? You need to make a payment of energy. So for example, if you are traveling in the highway, you need to make a payment. No toll එකක් ගෙවන්න එපා. You have to pay a toll. So similarly, if you want to use this transporter, you have to pay a fee, that is energy. The other type of transport is passive. That doesn't require any payment, but there is a filtration process. Filtration means, for example, when you are entering your school, if when you are going through your school gate, you are filtered, no? So everyone walking in the street can't get into your school. No? You have to be wearing a uniform. You should look like a student. Then you will be allowed to enter the school. But are you going to make a payment? No. At the school gate, sali ganne. Prefect lehema gannawad danne. Okay, right? So that is a passive transporter where you don't need to make a payment, but also you are filtered. Then you are going to write here, okay, මෙතන අපි ලියමු, provides hydrophilic channels for selective movement of substances. Don't go into the next diagram, okay? Don't go into the space of the next diagram. Okay, don't go into the space of the next diagram. Provides hydrophilic channels for selective movement of substances. Active proteins also filter, yes, because in the highway toll gate, motorcycles, three-wheelers are not allowed to enter. No? So think of everything, remember everything using examples, okay? So this is a ATP1 is active transporter. This the other one that is not requiring any payment. It's a passive transporter. Okay. Next one, enzymes. Now you can see here some of these proteins are acting as enzymes. Right? You can see a molecule that is going here, then it is coming out, then it is going here and coming out as an entirely different molecule. Right? That means there has been some reaction happening. So that has been triggered or let's say catalyzed by this protein molecule that is acting as an enzyme. Right? So in your resource they have given an example for these enzymes. That is microvillus on epithelial cell lining of some part of the gut contain digestive enzymes in their cell surface membrane. Okay, so now listen, when you eat some food, most of your digestion happens in your small intestine. In your small intestine, there are some enzymes built into the plasma membrane of those cells. There are some enzymes built into the plasma membrane of those cells. So those are called as bound enzymes. Okay, so that means they are sort of built into the digestive system cells. So we will say bound enzymes. Where are they found? Microvillus. I'll show you some microvillus. Okay. Microvillus on epithelial cell lining of some parts of the gut. Write down Taggala. I'll show you some what do you mean by microvilli, Harida, after we have completed this. Then this diagram is showing protein acting as receptor molecule. So you can write so new people, you will need to understand these are not theory teaching sessions. These are simply there. So main function of these up to speed sessions is to help you complete the tute. Alright. Then from there onwards you can join with our normal theory class as you without any worry that you started later. No, that's my main goal. Okay. Act as receptor molecule. So you can see this is the protein receptor. Meme cheese like a happy cow cheese. That is a receptor molecule. Could be a hormone. Could be what else? Could be a neurotransmitter. Neurotransmitter you have heard, no? Serotonin in your brain, dopamines in your brain. Those are signaling molecules in your nervous system. Okay, so they can bind with certain neurons, like nerve system cells, and they can have their own certain signaling activity. Okay, signaling activity could be there. Okay, act as receptor molecules. Then I want you to write down the receptor molecules that you have. Could be hormones. For example, when you achieve puberty, some of the testosterone like hormones in boys, there are testosterone receptors in your muscles so that the muscles get bigger. Okay, so the hormones can have an action on certain cells as well. Then write down the three types of these molecules: hormones, neurotransmitters, immune proteins. So once they bind, there will be a signal going into the inside of the cell. Then this next one is protein acting in cell to cell recognition. What do you mean by cell to cell recognition is in your body, your cells should be able to identify yourself. For example, if you were stopped by the police while you were driving a vehicle, you should be able to show your driving license and your identity card, no, to show that okay, I'm qualified to drive and I'm a Sri Lankan citizen, age above 18, so I can drive කියන එක. හරි. So for example, in your body there is a police, no, there's a police in your body, that is your immune system. So if your immune system detects things, one of your cells is not a body cell, it will destroy you, ne? So for example, if there are bacteria in your blood, the immune system is going to detect that. That is because the bacterial cell doesn't have the identity card of one of your own body cells. So the bacterial cell can't prove that to the immune system, mama is one of your own body cells. That's why the bacterial cell is going to be destroyed by your immunity. Okay, so your body cells, they have these sort of identity cards on their surface of their cell membrane, that is going to help them to identify themselves. They are similar to identity cards, identity tags. So in your school you have a name badge, no, saying you are from this school. Okay, එතකොට ගුටි කන්නත් වෙනවා from your rival school during big matches, නේ. Okay, when you have your school name badge. Okay, so cell to cell recognition usually by glycoproteins. I think there's a glycoprotein. What is actually the glycoprotein? The glycoprotein itself is actually this green color part, ne? The glycoprotein is actually this green color part. This is the protein. This is the antenna like carbohydrate. Then I'll skip this for now. I'll skip this. Okay, now this one is interesting. Okay, this one is interesting. This final one will talk about this structure that is called as the cytoskeleton. The cytoskeleton, as the name says, it is like the skeleton of your cell. The cytoskeleton is like the skeleton of your cell. So that is there to provide some support, like your own skeleton. It is there to provide some support. This is the cytoskeleton. Can you see? Inside of the cell there are some fibers. Okay, so these proteins are actually connecting to that cell, sorry, cytoskeleton, so that the plasma membrane itself has some support. Okay, this is also another supportive structure that you have outside the cell, that is known as extracellular matrix. That is also we have in the syllabus. Alright, so we'll label this. This is known as extracellular matrix. This inner structure is called the cytoskeleton. You know, cyto means anything to do with the cell, no? Cytoskeleton. Extracellular matrix and cytoskeleton. Then I want you to write help to maintain cell shape. Then this is also another way of cells maintaining shape, that is known as intercellular joining. That is not given in your resource, but I just kept that diagram because it can sometimes happen. Cells are connected by proteins. Cell and cells are several cells can be connected via proteins, so that they can form a tissue, right? So these two proteins are like, in Lego you can connect one building block to the other one. They are sort of connected with each other using this protein. Okay, so complete this. The entire set of functions of proteins. Taggal, I'll give you 1 more minute. You can either take a screenshot. Because I want to move on to the nucleus as soon as possible. Okay. Okay. Okay, now let's see. What do we mean by a microvillus? Okay. Intercellular joining. Okay, intercellular joining. Yeah, any question? Zoom in, it's not clear. Where do I zoom in? So I zoomed out so that everyone can see everything. I mean, from the top. Okay. We have to go fast. We have to go fast. I'll be sending this recording. You don't worry if you miss anything, Hari. But එමයි කියලා live එකෙන් යන්නත් එපා. Otherwise, I know you won't be going through the entire recording. Just keep in mind where you miss something and you can watch it later in the recording. Hari? Okay, now I can show you some of the things that I need to show using the software. Okay, right. So if you take a cross section of your small intestine, okay, you have taken a piece. It is very important that your small intestine has a lot of surface area. Okay, that is because the food digestion as well as the nutrient absorption has to happen there. Okay, so it is important that you have a lot of surface area. So you can see when we have folding, circular foldings. Can you see here rally, right? Right, waves and waves of tissue that is there to increase the surface. So if you zoom into one of these, you are going to see, not here, this sort of an appearance. These are known as finger like projections. These are called as actually villi. These are actually called as villi. Okay, so villi are all, I mean, these are actually macroscopic. You can see them with your naked eye. These villi you can see, right? The microvilli are actually found on the epithelial cells. They can, on the surface of in the surface of villi you can see cells with microvilli. Okay, so these are the cells that have microvilli. So they are not actually shown here, but these cells are going to have hair like projections that are known as microvilli, giving them an appearance like a brush. Brush board. Okay, so villi have smaller, I mean, on the surface of villi you have cells. No, these are the villi cells. On them you have microvilli. Okay, all right. So the next topic we have to talk about is the nucleus. Okay, so I'll show you how the nucleus is arranged. Now listen, if I remove this plasma membrane and remove some of this dark color cytoplasm, right, now you can see where is the nucleus. The nucleus is hidden by a large amount of membranes. Inside your cell you have a large amount of membrane that are actually very closely associated with the nucleus. Now if you take, this is an animal cell, no? In this animal cell, the nucleus is actually the centermost organelle. What is the, I mean, found at the center. So where is the nucleus? It is actually hidden, no? Anyway, small purple peeking out, that is the nucleus. That is because the nucleus is surrounded by a complex network of membranes. The outermost one is this. This is known as tubes, branching tubes. That is the smooth endoplasmic reticulum. SER, smooth endoplasmic reticulum. Then inner to that you have this flattened, flat network of membranes. These are called as rough endoplasmic reticulum. This is the rough endoplasmic reticulum. So if you remove this rough endoplasmic reticulum, if it you can see it as a cross section, or you can see it as a cross section because there you have sliced it. Now you can see the nucleus. It's like a circular stadium, right? Maybe something you have in maybe a football stadium. Okay, this is like the roof of a such a stadium. Okay. So this is the structure of the nucleus. The nucleus is actually bound by two membranes. Both membranes together form what we call as the nuclear envelope. So the nuclear envelope has two membranes actually. There's an outer membrane and an inner membrane. Okay. Then this is the inside of the nucleus. This is the inside of the nucleus. And you can see the rough endoplasmic reticulum, which is this. The rough endoplasmic reticulum is actually connected to the nuclear envelope. The rough endoplasmic reticulum is connected to the nuclear envelope because we say it is actually continuous. We actually say, you can say the outer membrane of the nuclear envelope. Nuclear envelope can the covering of the nucleus is going to become the rough endoplasmic reticulum. Right? Is going to become the rough endoplasmic reticulum. So inside the nucleus, we are still talking about the nuclear envelope now. So you can see in the nuclear envelope, there are small openings. There are small openings. Those are called as nucleopores. Can you see? Right. Those are called as nucleopores. So things from inside the nucleus can come out through those openings. Those are called nucleopores. Nucleopores. Okay. Then what else? So we'll come to the inside. In the inside of the nucleus, there's this dense area. Not like a center. It's not always at the center like this. Right? There's a dense area that we call as the nucleolus. It is actually a production area. Mainly it produces ribosomal components. Mainly ribosomal rRNA is produced in this nucleolus. Then you can see some of the chromosomes that are formed out of the DNA can be seen. Then this is the inside of the nucleus, which we are going to learn in some more detail. This is the inside of the nucleus. Okay, clear? Any other questions? That's just a rough overview. Then I'll quickly show you from there. From the rough endoplasmic reticulum, the smooth endoplasmic reticulum arises. From the rough endoplasmic reticulum, you can see the rough endoplasmic reticulum is this flat tube-like flat membrane. Then from that you get a tube-like network. This tube-like network, that is the smooth endoplasmic reticulum. Both of those we are going to learn very soon. What else can you see in this cell? This is this green color one, that is the Golgi apparatus, mainly Golgi. And it is a mitochondria. This red one, that is a mitochondria. There's another mitochondria here. This is a peroxisome. We'll talk about those peroxisomes very soon. Then there's a lysosome here, I think. This is a lysosome. This yellow color again. These are lysosomes. We'll talk about those as well. Those are the things. This is the centrosomes. Then centrosomes we'll also talk about. You can see nicely their microtubule arrangement. Okay. But we'll finish talking about nucleus first. No? Any questions that you want to ask? So we'll come to the nucleus. After doing nucleus, we'll take a small break. Correct? Nucleus ඉවර කළා මෙමු. හරි කට්ටිය නිදිමතයිද නෝ? When do you have dinner? දැම්ම කන්නේ නැහැ නේ. So I'm having dinner after finishing this session. So you guys can wait. Okay. Hurry. Come to the nucleus. Come to the nucleus. Yeah. The other black dots around rough endoplasmic reticulum, ribosomes. Yes, very correct. Yes. Nucleus. Hurry. We'll read. Come to the new resource book. Let's read it. So, page number 29, subcellular components. Let's read. There are many subcellular components in the cell. Some of them are organelles, which are bound by membranes and suspended in the cytosol of a eukaryotic cell to perform specialized functions. Highlight the definition of an organelle. Highlight the definition of an organelle. Highlight. Bound by membranes and suspended in the cytosol of eukaryotic cell to perform specialized functions. These are organelles. Okay. Organelles are bound by membranes and suspended in the cytosol to perform specialized functions. Right. So, nucleus. Let's read. Nucleus is the most prominent organelle, consists of most of the genes, having an average diameter of 5 micrometers. Can you highlight? The average size of the nucleus is around 5 micrometers. 5 micrometers. Enclosed by a double membrane called the nuclear envelope. Highlight. It is a double membrane bound structure, that is the both membranes together we call it as nuclear envelope. Okay, both membranes together is known as the nuclear envelope. Okay. Right. Then nuclear envelope again we'll read. Okay, so there are 4 components. You can see there are 4 dots in the resource book. There are 4 dots. 1, 2, 3, 4. Okay. So you can put number 1 in the first dot, that is about the nuclear envelope. Let's read. Composed of 2 membranes, inner and outer, separated by a space of 20 to 40 nanometers. Highlight. It has 2 membranes. So you can see in between, when these 2 membranes, you can see there's a space, no? You can see there's a space in between these 2 membranes. Can you see? I hope in your tube diagram it is clear. There's a small space, no? That is the intermembrane space. Highlight the width of it, 20 to 40 nanometers. 20 to 40 nanometers. Okay. Nuclear envelope is perforated by nuclear pores, which has pore complex to regulate the entry and exit of substrate. Highlight. Nuclear envelope has perforated by nuclear pores having pore complex. You can see some of the nuclear pores, maybe. Nuclear pores. Highlight pore complex. Okay. Pore complex is actually, it's like again like a toll gate. It's like a it's like a gate, and it's composed of a certain ring of proteins. It is composed of a ring of proteins that can regulate the things that are trying to get into the nucleus and also going out of the nucleus. Okay. Pore complex, please highlight. Then it has nuclear lamina, which made up of protein filaments that line the interior side. Highlight. Nuclear lamina made up of protein filaments lining the interior of the nuclear envelope. Very important. What do you mean by a lamina? Then if you take a look, you can take a look at your roof. If you have a slab, then that's fine. Take a look at your roof, that is mainly made up of wood, no? Wooden beams, they are crossing, going in every way, right? So what is that doing? It is supporting the structure of the roof, no? So if you have been into the inside of an indoor stadium or that sort of a big stadium, if you have been inside, you can see there's a complex network of metal bars that are supporting the curved roof, round roof supporting. Yeah. So the nuclear lamina, keep this in mind, the nuclear lamina is something like that is supporting the nuclear envelope. It's a protein filament network supporting the nuclear envelope. Okay. Then okay, next dot, Harry. made up of protein filaments. Nuclear matrix, highlight the word nuclear matrix, made up of protein filaments and extend throughout the interior of the nucleus. Highlight, made up of protein filaments and extend throughout the interior of the nucleus. Chromatin and nucleolus are embedded in the nuclear matrix. Okay, so I'll explain what is chromatin. So nuclear matrix is everything that you have inside the nucleus itself. So now you can, in your house you have these columns. There are walls that are supporting the roof itself, So the nuclear matrix is the interior supporting structures of the nuclear envelope that are found inside. The nuclear lamina is lining the inside. They can sort of attach to the inner surface. You have the nuclear lamina. The nuclear matrix is found actually in the inside. So whatever you have inside the nucleus is actually embedded in the nuclear matrix. Okay. Next, nucleolus appears darkly stained granules with fibers in adjoining part of the chromatin. So if you sort of want to observe the nucleus under the microscope, the nucleolus, that is sort of the center part, is actually the most dark looking structure. It looks very dark. Okay. Then finally, chromatin appears as a diffuse mass. Can you highlight? Appears as a diffuse mass in electron micrographs of non-dividing cells. This phrase non-dividing cells is very, very important. Why non-dividing cells? Because in a non-dividing cell, the DNA is not properly packaged. For example, when you are studying, you don't pack your books into like separate bags every time after you have completed studying. Or let's say if you have your everyday clothes, you don't always tightly fold them and put into a travel bag, because why? You use them every day. But if you are about to travel, you pack your clothes very tightly, very neatly into a suitcase or a travel bag, right? So similarly, when a cell is not dividing, when it's normally a cell, he is not going to have his DNA tightly packed into a chromosome. He is going to have it in the form of a thread-like structure that we call as chromatin. I'll show you how chromatin is formed. Okay. So that's why we say it acts as it is visible as a diffuse mass. That means diffuse means a sort of loosely arranged in electron micrographs. During nuclear division, chromatin condenses, tightly coils and forms threads called chromosomes. Each species has a constant number of chromosomes. Okay. Can you highlight? Chromatin appears as diffuse mass in non-dividing cells. Diffuse mass in non-dividing cells. Diffuse mass in non-dividing cells. Then highlight each species has a constant number of chromosomes. So you as a human, you should have every one of your cells except what cells? Every one of your cells should have 46 except which cells? If you are a man, what cells don't have 46? Sperm cells don't have 46. They have only 23. Female, your eggs don't have 23. Sorry, 46. They have only 23. Right. Now let's explain all this. Okay, we'll make a note here. Nucleus. Okay, now listen. I want you to write down average diameter 5 micrometers. Average diameter is 5 micrometers. Then we are going to write down each of the components. Nuclear envelope, has nucleopause with O complex. These diagrams, there are some words around these diagrams. They are not just there for you to chill just for the tute to be there. Okay, you have to read them. Okay, they are important things that are directly from your Campbell biology. Campbell biology is your direct resource book. Your resource book is made up of Campbell. So I'm putting these diagrams so that you don't have to waste time going through the Campbell. Campbell එක බලන්න ඕනේ නැහැ. Got it? Because I'm going to put whatever the diagrams you need in the tute. You just have to study the tute. Sorry, tute means the resource book. I mean, everything else you need, everything extra that you need is going to be in the tute. Okay? And the nuclear envelope, you can say, there's an outer membrane and an inner membrane separated by a 20 to 40 nanometer space. That is all about the nuclear envelope. Second structure. Wait, hold on. Put a star mark here and write down under the nuclear envelope. Nuclear lamina composed of protein filaments line the interior of nuclear envelope. Second one, nuclear matrix. Extend throughout the interior. Also made up of protein filaments. Okay, you write properly. Also made up of protein filaments. Also made up of protein filaments. 3. Nuke darkly stained granules with fibers. Synthesize rRNA, ribosomal RNA. Last week, okay? Synthesize rRNA. Fourth one, chromatin. Chromatin we'll write down here, I'll explain, okay? Chromatin is equal to DNA plus histone protein. I'll explain this, okay? Chromatin is equal to DNA plus histone protein. When chromatin undergoes condensation, we call that when chromatin undergoes condensation, we call that chromosomes. I'll explain what is chromatin. Nuclear envelope. Okay, I'll read whatever I have written here. Nuclear envelope has nucleopause with the pore complex. There's an inner membrane and outer membrane separated by a 20 to 40 nanometer space. Nuclear lamina is composed of protein filaments line the interior of the nuclear envelope. Then second component is nuclear matrix. Extend throughout the interior, composed of protein filaments, also composed of protein filaments. Nucleolus, darkly stained granules with fibers, synthesize rRNA. Okay, nucleolus, darkly stained granules with fibers, synthesize rRNA. Chromatin is equal to DNA plus histone proteins. Chromatin undergoes condensation to form chromosomes. Okay. Can you read the chromatin? Chromatin is equal to DNA plus histone protein. Histone. Histone. It undergoes condensation to form chromatin. Okay, now listen. Histones are something like this. Histone proteins have a composition. Can I go to the whiteboard? Yeah. Whiteboard means make a whiteboard deck. Okay. Okay, so condensation of DNA. So you know DNA has this Mehema structure. So we talked about the DNA structure last week, no? Right? It has our nice double helix structure. So, but this double helix, you have so much length of DNA, we have to find a way to pack this DNA into the limited amount of space that you have inside the nucleus. I mean, okay, so that is done by using a special packaging method using a bowler protein, a spherical protein that we call as histone. So actually 8 histones. The method you can see 1, 2, 3, 4. There's another 4 at the back. Okay, so you take 8 of these histone proteins, and what you do is you wrap DNA 2 times around this 8 histone complex. Then again you have the DNA molecule going. Again you take another set of 8 histone proteins and you wrap the DNA around that as well. Okay, so again you go. So this arrangement of DNA is what you have in non-dividing cells. That is called chromatin. Just DNA wrapped around histone. Simply DNA wrapped around histone. But this is going to undergo a lot of condensation, right? All this DNA and histone is going to undergo a lot of condensation. It's going to fold like this. Then itself is going to fold like this. And at the end, you are going to get a very dense structure containing all the entire DNA molecule condensed into a very small structure that we call as chromosome. That is what we call as chromatin undergoing condensation. You can remember it's happening in prophase of cell division. If you haven't done prophase, we'll be doing in up to speed sessions. So you don't need to worry about that. Okay, so this is nucleus and how chromatin is formed. We'll talk about this again when we are doing cell division. Okay, clear about nucleus? Okay, you can have a small break. Then we'll move on to ribosomes. We will talk about endoplasmic reticulum, lysosomes, cytoskeleton. A lot of things are there to be talked about. Okay, so taggalenna. Many things are need. Many things. I mean, we have a lot to cover today. Are okay? Taggalenna gilla. We start again at 8. කියවන ගන්න 8:20. Okay, small break. Taggalin. Okay, all right, we'll continue then. Okay, come back, come back. All right. Okay, shall we move on to ribosomes? Yep. Let's move on to ribosomes. Okay, come to the resource. Page number 29 still. Okay. Hold on. There are some some things about the functions of nucleus. Let's read. Control all cellular activities, of course. The nucleus acts as like the cockpit or the control center of the entire cell. Okay. Synthesize DNA to produce new nuclei for cell divisions. We'll talk about the cell cycle next week. There's a phase of DNA making before the cell divides into so that is S phase. So that DNA making occurs inside the nucleus itself. Synthesize rRNA and ribosomal subunits required for protein synthesis through the nucleolus. Can you highlight synthesize rRNA through the nucleolus? Then synthesize mRNA and tRNA according to the information present on DNA. Okay. Synthesize tRNA and mRNA as well. So mRNA as well as tRNA production occurs in the nucleus as well. Okay. That's info. Okay. Finally, store and transport genetic information. Yes, it's act as the information library for all the proteins that are going to be produced in your body. Proteins include enzymes, immune system proteins, then other structural proteins. You know all the other functions of proteins. Okay, all those are made by looking at the information present in the nucleus. Okay. We'll come to ribosomes. Okay, so this chart, this box we are going to fill up with the ribosome knowledge. You have two types of ribosomes that you need to know, 70S and 80S kira. Okay, so let's read page number 29. App you are using to show 3D diagrams. Yeah, it's called visual. For anatomy, it's an app I used to have in my first year in medical faculty that everyone had. That is 3D human anatomy or something. Then the other one actually bought when I started doing classes. That is called 3D biology or something. Yeah. The other one is called Anatomy Atlas, and this one is called just biology. Okay. Anyway, 29 ribosomes. These are subcellular components which carry out protein synthesis. Subcellular components which carry out protein synthesis. Highlight. If they ask you what is a ribosome, they are subcellular components. Now listen. Why do we call them as subcellular components? We highlighted earlier, to be considered as an organelle, you need to be usually bound by a membrane. You need to be bound by a membrane to be considered as an organelle. Okay, so these are simply subcellular components. Subcellular means what? You find them inside the nucleus. That's it. Okay, you find them inside the nucleus. Highlight. Okay, then keep reading. They consist of two subunits, a larger subunit and a smaller subunit. A ribosome has two parts, a larger subunit as well as a smaller subunit. Then they are composed of rRNA mainly, and then some protein, other parts could be there. So highlight, they are composed of rRNA and protein. So if you take a ribosome, they have a typical appearance composed of two subunits. There's going to be there's a larger subunit like this. In your molecular biology lesson, we are going to talk in detail about the structure of ribosome. So this is the larger subunit, this is the smaller subunit. For every ribosome, you have these two parts. Ribosomes are of two types, as 70S and 80S. Then read, 70S are found freely on the cytoplasm of prokaryotes, matrix of mitochondria, and stroma of chloroplast. Right? Now listen, 70S ribosomes are usually smaller. This S S is something short for sedimentation coefficient. You don't need to know that. Because when you sort of centrifuge, you rotate your cells to extract your cellular structure. So when you rotate them, what we call as centrifugation, the cell is going to be broken down and cellular structures are going to come out. Then when you allow them to settle, okay, or what we say as sedimentate, sedimentation, settling down under their weight. So what is going to settle down faster? The big one, no? So by the rate of sedimentation, the way they settle down, you can classify them according to size. So your smaller one has a sedimentation coefficient of 70 and your larger 80 they are larger because their sedimentation is higher. Okay, so actually it's a sort of a running joke, right? These larger and smaller subunits of a 70S ribosome actually has 50S large subunit and 30S smaller subunit. I know it is 80, but because when they are together, they are more compact in the, we say 50S and 30S form 70S. Okay, if you take your 80S, it is actually the larger subunit is 60, smaller subunit is 40, but altogether we say they form 80. Right? That is because when the subunits are together, they are more compact. The overall S value goes down. Okay. So anyway, you don't need to know this subunit values. You don't need to know. You only need to know there's a larger subunit and a smaller subunit. Got it? So 70s are smaller, 80s are larger. Then you need to know where are they found. 70S ribosomes are only found in this list of locations. They are found in prokaryotes such as your bacteria, right? Prokaryotes like then whereas inside the eukaryotic cell there are only two locations where you have 70S. Inside the eukaryotic cell you have two locations. One place is in the mitochondria, in the sort of the gel-like part of mitochondria known as matrix of mitochondria. We'll learn about it soon, okay? The inner structure of mitochondria. And also similarly, they are found in the chloroplast, particularly what we call as a jelly-like material inside the chloroplast, that is stroma of chloroplast. Okay, then 80S ribosomes, you need to know they are only found in, okay, put a star mark and write, only found in eukaryotes. Then there are two types of 80S based on how they are organized. Okay, you can have them as free ribosomes. So they are found in groups. Free ribosomes are just randomly found in groups in the cytoplasm of the eukaryotic cell. They are just found randomly in the cytoplasm of the eukaryotic cell. Then there's another form of 80S ribosomes in your eukaryotic cell that we call as bound. Can anyone tell me where do you have the bound ribosomes? Yeah. Attached to rough endoplasmic reticulum, or you can write RER to save space, but paper don't write RER, you have to write the full name, rough endoplasmic reticulum. Okay. Okay, can you complete this? 70s are smaller, found in prokaryotes, matrix of mitochondria. So these are eukaryotes, either. matrix of mitochondria of and stroma of chloroplast of eukaryotic cells. ATS are larger and they are only found in eukaryotes. They are of two types, as free, found in groups in the cytoplasm, bound, attached to rough endoplasmic reticulum. Hari? Okay. Then there's some more to be read in the resource book. Can you jump to the resource? ATS ribosomes. Next page, page number 30. Okay. ATS ribosomes are only found in eukaryotes. Based on their presence, ATS ribosomes, did you highlight? Found only in eukaryotes. Okay. ATS are only found in eukaryotes. Based on the nature of their presence, ATS ribosomes are organized as two types, free and bound. Okay, free are available as groups in the cytoplasm. Bound are attached to the membrane surface of rough endoplasmic reticulum. Okay, a lot of MCQ points could be there. Okay, MCQs could be asked so much from these. Therefore, it's very important that you have a nice idea about all the, because what I am always trying to do here is to organize the information that you have in the resource so that it's easier for you to organize it in your brain. Okay, so if you just have it as paragraphs, you are not going to memorize it. No? So what I'm always trying to do is sort of organize it into sort of memorizable pieces of information that you can easily get into your memory. Okay, then we have to move on to RER. Any questions? Any questions? Going too fast? No, right? Oh, the chat is very inactive. Okay, shall we move on to lysosomes? Sorry, endoplasmic reticulum. Okay, let's read. I think I already showed you the endoplasmic reticulum using the software, right? Hurry, Abhi, let's give it a read, and there's some things to be highlighted. Okay, chill ke karne. Endoplasmic reticulum, it's a network of internal membranes. It's a network of internal membranes forming flattened or tubular sacs. All right, so you can see in this diagram. Okay, so what's missing from here is the nucleus. What's missing from here is the nucleus, and you can see, as I mean, it is continuous with the rough, I mean, outer membrane of the nuclear envelope is going to immediately give rise to the rough endoplasmic reticulum. Ne? So you can see what are these mal in the nuclear envelope? Yeah, what are those? Anyone? What are these? These structures? Nucleopores, yes. Then, okay, so you can see the rough endoplasmic reticulum arising out of the outer membrane of the nuclear envelope, and it is, you can see, it is all these bound ribosomes. You can see all these bound ribosomes in the rough endoplasmic reticulum. Then from the rough endoplasmic reticulum outer edges, you can see the SCR that is arising. That is the outermost part of the endoplasmic reticulum itself. Then you can see transport vesicles are being, coming out of the endoplasmic reticulum. That is actually one of the functions of endoplasmic reticulum, to produce these transport vesicles. Okay. So what we are going to do is, we are going to do a nice comparison between the two types of endoplasmic reticulum in this chart. Okay. All right. Let's read about ER. Definition is there. It's a network of internal membrane forming flattened or tubular sacs separating the cytosol from the ER lumen. So everything that you have outside this membrane of the endoplasmic reticulum is actually the cytoplasm. Right? It is the cytoplasm, or the jelly-like material inside the cell. So what the endoplasmic reticulum is doing is, it has a set of membranes that is separating the inside of this endoplasmic reticulum from the cytoplasm of the cell, so that it's a separate inner membrane unit. Okay. And it is continuous with the outer membrane of the nuclear envelope. Highlight. It is continuous with the outer membrane, as you can see here. Okay. It is continuous with the outer membrane of the nuclear envelope, which is a very important point that they can ask in the MCQs. There are two types of ER, as rough ER and smooth ER. Rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough ER consists of flattened sacs with ribosomes bound to surface. Highlight. Rough ER is flattened with ribosomes bound to surface. So you can write on the RER. We'll take this side as the RER side. We'll take this as the SCR side. You can write on the RER side, they are flattened with bound ribosomes. On the SCR side, you can write SCR is actually tubular, bata, right? Tubes, okay? You can have bound enzymes, not ribosomes. You can have enzyme molecules bounded to the surface, okay? Okay, keep reading. Proteins synthesized by the ribosome move into the lumen of the ER. Rough ER, one of the main functions of the rough endoplasmic reticulum is protein synthesis. Because of why? All these ribosomes make the protein. Okay. So many functions are there. So from this point onwards, we'll write down the functions. Sorry, listen. What are the functions? There's a list of functions that I want you to remember. Okay, very carefully. Okay. One, transport proteins synthesized by ribosomes. Then the other synthesizing functions are there. Synthesize glycoproteins. What else? Synthesize also transport vesicles. And it's like a vehicle manufacturing plant. The transport vesicles are like transport lorries. They can be loaded up with anything. You could be newly made proteins, could be newly made enzymes. Vesicles could be loaded up with things to be transported, and they could be sent out. Okay, so RER synthesized glycoproteins, transport vesicles. You can write membrane phospholipids. Membrane phospholipids and proteins making, that synthesis of membrane phospholipids and proteins is a very important function of the rough endoplasmic reticulum. Therefore, you call it as the membrane factory. Now listen, the plasma membrane as well as all the other inner membranes inside a cell need the raw material. When you are making them, you require some sort of raw material. What are the raw materials? Phospholipids and protein. So those raw materials are synthesized by rough endoplasmic reticulum. Okay, so that's what they say. Okay, come back to the resource book. Produce transport vesicles, synthesize membrane phospholipids. Okay, then very important statement there. Facilitate the growth of own membrane by adding phospholipids and proteins and carbohydrates. Therefore known as membrane factory. It's a nice word that you have to always use if they ask you in the structured essay paper, probably ask you to write down what are the functions of rough endoplasmic reticulum. You have to say act as a membrane factory. Important function. Okay. අපි SCR ලියලමු. Okay, then what are the functions of SCR? You can say functions of SCR, you can say synthesize lipids. That's an important function. Synthesize lipids. What are the lipids? Oils. If it's a plant cell, a lot of oil can be produced by the SCR, smooth endoplasmic reticulum. Then steroids. Steroids are what? Usually cholesterol based. Steroids can be cholesterol based things. Then phospholipids can also be synthesized in the smooth endoplasmic reticulum. Then very important statement. I want you to right? You have to pay a lot of attention. Produce transport vesicles, but listen. These are only for local transport. That means only for be transported within the cell. Okay, only to be transported within the cell. So you can say transport vesicles within the cell. Then there's another important function that you can write. Detoxification. Okay, detoxification means whenever your cell has absorbed some toxic substances, can you tell me what is the organ in your body that has to deal with the almost all the toxins in your body? All the toxins, which organ in your body has to deal with that? All that poisons, toxic molecules, highly reactive molecules are the liver. The liver cells or the hepatocytes have a lot of smooth endoplasmic reticulum that is there, so that there are enzymes there to break down these toxic molecules. Okay, so SCR is involved in detoxification. Then there's another important function, calcium 2+ ion storage. Calcium 2+ ion storage is a very important function done by smooth endoplasmic reticulum. That is happening in not in every cell. Every cell doesn't require calcium. Okay, calcium storage is required in Sarcoplasmic reticulum. Okay, I'll pay careful attention to the spelling, okay? Sarcoplasmic reticulum. There is a special type of smooth endoplasmic reticulum known as sarcoplasmic reticulum in muscles. So, your calcium has to be, calcium is a very important ion for the contraction of muscle. Therefore, it is stored in a special type of smooth endoplasmic reticulum called sarcoplasmic reticulum in muscles. Okay, so we'll quickly finish what is there in the resource, then you can quickly take this down. Okay, produce transport vesicles to transport within the cell. That is a function that I told you. Inulin detoxification, important function. Stores calcium ions. You have to write down calcium 2 plus storage in the sarcoplasmic reticulum in muscles. Harry, can you quickly take this down? Okay. Any questions? Shall we move on to lysosomes? Any questions? Oh, Katti, the chat is very dead. After dinner after dinner, all the energy is gone. Okay. We'll go ahead. Okay. Then we have actually Golgi. Golgi, we don't have anything to write down actually because this diagram is sufficient and all the things that you need to know are given in the resource. Okay, so I want you to follow this diagram as you highlight the resource. Then we'll, for a change, we'll go and do a question. Hari? Just doing theory parts. No? Hari, we'll read about Golgi apparatus, Stuck page number 32. Okay, Golgi apparatus is a stack of flattened sacks or cisternae. Okay, stack of flattened sacks. These are Golgi, you can say. So these levels, all these membranous structures, these are called as cisternae. Can you highlight? If they ask you what is a Golgi, you have to say it's a stack of flattened sacks. Those flattened sacks are called cisternae. Okay, so that's how a Golgi is composed of. Right, inner and outer surfaces can be identified as cis and trans phase. Okay, there's an inner surface, or we call this, you can write in this diagram if you want, it is known as the receiving phase. How is the cis phase is known as the receiving phase. So if you go to a post office, have you ever gone to a post office? In the post office, there's a site in the post office where you hand over the parcel or the letters. Parcel there's a site where you have to hand over your parcels or letters. So in a Golgi is like a post office. So similar to a post office, in the Golgi, there's a site that we have to hand over the vesicles that are coming from the nucleus, not nucleus, but from your endoplasmic reticulum, rough and smooth, will bind with the Golgi at the cis phase. CIS, cis. Okay, then the things that are inside those vesicles, they will go through all these layers of membrane similar to a post office. So if you give a parcel to a post office, they will check the address. Okay, this has to be gone to this district and this area in this district. So they will categorize. No, they will categorize and reconfigure where they have to go. Okay, so that is going to happen inside the Golgi as well. So they will go through all these levels, and afterwards, when they have been repacked or they are ready to go out, they are going to be going out from this phase. This is known as the trans phase. Sorry. So keep reading. Cis phase is located near the endoplasmic reticulum to receive vesicles from ER. Okay, located near the ER. You can see this is the endoplasmic reticulum to receive vesicles from ER. Trans phase give rise to secretory vesicles. Highlight. Trans phase give rise to secretory vesicles, which are budded off and travel to the other side. Golgi apparatus is abundant in secretory cells. Secretory cells could be certain cells in the liver have a lot of secretory function. Okay, certain cells in the kidneys have a lot of secretory function. Could be maybe in your pancreas, there's a lot of secretory cells producing enzymes or whatever. Pancreas, Agnashya, right? Where you produce a lot of digestive enzymes. Okay, so those kind of cells have a lot of Golgi. Then very important functions: collecting, modifying, packaging and distribution of materials, mainly proteins and membrane lipids. Can you highlight those words? Collection, modifying, packaging, distribution. Exactly like a post office. You collect, you modify, you again repack, and then you distribute. Okay, that's what happens in Golgi. Okay. Anyone else who's not in the groups or didn't get the tune? Quickly tell me. Okay. Anyway. Okay. Anyway. Then the next function is such an important one. Manufacturing cellulose and non-cellulose cell wall components such as pectin. Highlight that. Okay. So your cellulose and the other things such as hemicellulose as well as pectin that are necessary to make a new cell wall, that are necessary to make a new cell wall, exactly like this. Where do you have a new cell wall being produced? There is a situation that we call as a cell plate being formed. Okay, a cell plate being formed. So when a plant cell is ready to divide into two plant cells, you can see this is the old plant cell, and there's going to be a new cell wall produced that are from, that is, I mean, this new cell wall is going to be produced from certain vesicles. You can see in this electron micrograph, you can see here the vesicles are forming. Alright, it's like you are building a wall by piece, and later you are going to connect all the pieces of the cell wall to form the continuous cell wall. Okay, that process is known as a cell plate formation. The cell plate. Okay. So after the cell plate has been completely formed, you are going to get a nice new cell wall. So all these vesicles that are forming this new cell wall is going to come from here. Come from where? Golgi. It's going to come from Golgi. Okay. So in your resource book, I want you to write down. In your resource book, I want you to write down. If you don't like writing in it with a pen, you can write with a pencil. Okay. You can write there. Manufacturing cellulose and uncellulose components such as pectin, right in front using an arrow, forming cell plate. Forming cell plate. Okay. Forming cell plate. The cell plate is going to become the new cell wall actually. Cell plate taking the cell wall is going to arise. Then next function, produce lysosomes, which is the next topic that we are going to discuss. Just to sort of so this is not how I usually do in our theory class. I usually do questions in between, but because I'm going a bit rapidly, we don't have much time to do the questions. Okay. So can we do a few questions from yeah. Can you do this one? Question number 6, Taggalakan, I'll give you 1 minute. Taggalakan one, question number 6. Okay, send me answers, everyone. What do you think? It is mainly composed of carbohydrates, phospholipids, and proteins. No, neither. So you can correct this using a red color pen or something. You can cut the carbohydrates. It's mainly composed of phospholipids and proteins. Right. Phospholipid molecules are movable and provide a fluid nature. Yes. Out of the fluid mosaic model, the fluid nature is given by the plasma membrane. Can anyone tell me mosaic nature is given by what? By the proteins embedded randomly. Proteins embedded randomly. Peripheral proteins are tightly attached to the outer surface? No. You can say they are loosely attached. You can cut this and say you are loosely attached to the outer surface. Phospholipid bilayer enables nearby cells to communicate with each other? No. It is wrong. You can say proteins embedded. Neither. You can actually write a small review here. Proteins embedded in the plasma membrane. Proteins embedded in the plasma membrane identify the cell. Identify the cell. Finally, hydrophobic tails of phospholipids attach to cytoskeletal fibers and help to maintain. No. Right? There is again some proteins. Neither again it's something, it's a protein that's going to attach to cytoskeletal fibers. Some proteins attach to cytoskeletal fibers. Hurry. Okay. Yeah, so the answer is answer number 2. Neither. Looks good. Okay, shall we come back to our tute? We have to talk about lysosomes. Okay. Lysosomes. So lysosome perform some of the most important functions and they are very dangerous as well. They are dangerous weapons that can harm a cell as well. So they have to be very carefully maintained and regulated, otherwise they can cause damage to the cell itself. Okay, lysosomes. They are powerful. They are powerful weapons. Okay, so let's read. Lysosomes are single membrane bounded vesicles contributing to digestive activity. Highlight single membrane bounded vesicles. They have only one membrane. So, for example, if you take the if you take the nucleus, you have two membranes. So, single membrane bounded vesicles contributing to digestive activity. Okay, lysosomes. They contain hydrolytic enzymes which catalyze the breakdown of carbohydrates, proteins, lipids, and nucleic acids. Highlight, they contain hydrolytic enzymes. Hydrolytic enzymes means these enzymes add water. Why do you need to add water? For example, you know, a carbohydrate is formed by a glycosidic bond. How does a glycosidic bond form? By a dehydration reaction. You lose water. So, if you want to break down a carbohydrate, what do you need to do? You add water. Okay, and also peptide bond. A peptide bond is also a dehydration reaction formation. So you add water, you break down proteins into amino acids. Understood? So that's why these are called as hydrolytic. You break down using water. Highlight that they contain hydrolytic enzymes. Okay. So you can write here, put a point and you can write down single membrane bound vesicles with hydrolytic enzymes. Okay, then what are the functions? Digest food particles received by phagocytosis. Phagocytosis, also known as cell eating. Okay, in your body there are specific cells that are known as phagocytic cells. For example, cells of the immune system. They are designed to eat bacteria. Okay, so imagine you got a bacterial disease in your lungs. Okay, these type of phagocytic immune cells that are going to come into your lungs, they are going to eat those bacteria by the process called phagocytosis. So that's what's happening here. Imagine so they are giving using a small piece of food. Imagine this piece of food is a bacteria. So it is going to go through the plasma membrane, and that plasma membrane is going to make like a let's say a package, food package. Making a lunch box it's a parcel of food. Out of the plasma membrane, you are going to produce a food vacuole. We are going to learn about food vacuoles very soon. Okay, you are going to make a food vacuole. So here comes your lysosome, which is your weapon. It contains digestive enzymes, and they are going to combine, and the enzymes are going to digest the food particles. That's how phagocytosis works. Then once this food particles are digested, actually once digestion is completed, it's going to left out with some of these undigestible components. So after you had your lunch, you're going to have some of these bones of fish and these carapincha pieces. Those kind of things you are going to end up with. No? So those things are like waste. So all the undigestible things are thrown out. That is known as exocytosis. Residue material thrown out. Okay. Finally, digest worn out organelles. Sorry, not finally. The next one, worn out organelles. For example, maybe there's a mitochondria here. Imagine like it's a mitochondria is an organ that is producing energy. No? So after it has been producing or working for a long time, it could get damaged. So then it's no longer very useful. It's better to make a new mitochondria than using the same old one. Vehicle you put a new engine so that you are more efficient. So you can also break down that old organelle and use all the proteins and other things that you got to make a new organelle. That is the worn out organelles, digestion of worn out organelles. Then the final function is autolysis. What do you mean by autolysis? Auto means self. Tamangoma, right? Autolysis means when there's a defined very specific mechanism to do this. When a cell feels like the cell has been damaged beyond repair or it has reached the end of its life cycle, the cell is going to commit suicide. I have lived a long life. The cell is going to commit suicide. That is known as autolysis. For that, what you are going to do is, all these lysosomes that are found inside the cell, they are going to simply release their enzymes into the cell. The enzymes that are hydrolytic enzymes, they will be released into the cell so that the cell will be digested from the inside. He will be killed by his own lysosomes. That is known as autolysis. So I want you to write down the functions here. One, digest food by phagocytosis. 2, transport residue out. By exocytosis. 3, digest worn out organelles. Finally, autolysis. Those are the functions of lysosomes inside the cell. Okay, those four you have to remember one by one. They will ask definitely. Structure, they say particularly list out the functions of lysosomes. Okay. Then we have to talk about glyoxysomes. Okay. I'm sorry. Before that, yeah, okay. Sorry, peroxisomes. We have to talk about peroxisomes. Okay. So listen, peroxisomes ke we can write down a small subtopic here. Katti not bored neither, right? We have to go a bit fast, It's inevitable. All right. Then let's read about peroxisomes. They are single membrane bound vesicles with oxidizing enzymes. Okay, they are found in both animals and plants. Enzymes in peroxisomes catalyze the breakdown of H2O2. Hydrogen peroxide H2O2 is one of the most reactive molecules that you can have in your body. It is going to cause a lot of oxidative damage by reacting with sensitive molecules such as proteins. So as soon as you have some hydrogen peroxide in your body, it has to be broken down. Otherwise, there could be irreversible damage to some enzymes and other things. Okay. So therefore, peroxisomes are special task force that are designed to break down peroxisomes as soon as you have them. Okay. So you can say H2O2 will be broken down to oxygen gas and water by peroxisomes. Then you can say they contain oxidizing enzymes. You will do this when have you done redox reactions or not yet neither? Some chemistry classes they are doing the first lesson and the third lesson all together. එහෙම කරන chemistry classes තියෙනවා නේද? Right? Those people might have done redox reactions. So there you will do how hydrogen peroxide breaks down into oxygen gas and water, whatever. So for that, so this is going to be good, ne? So you have neutralized the hydrogen peroxide so that it's no longer toxic. Then keep reading. Functions, detoxification of peroxides and photorespiration in plants. So you guys have been joining the normal theory class, right? Anyone who didn't join the normal theory class yet? Because we finished photorespiration in the normal theory class. You guys have been, right? So if you enroll, in the website, you have this month's recordings. July month, I think I started with the end of enzymes. Ne? Enzymes final day, then we started photosynthesis day 3 was completed at Saturday. This Thursday photosynthesis day 3 at the Thursday batch. Okay, so photosynthesis, everything is there in the July month course in the website. So by then you know what is photorespiration and you know how peroxisomes are involved in photorespiration as well. Okay. So then detoxification of peroxides and photorespiration in plants. Those are the main functions. Then we are going to have another subtopic that is important to discuss. That is called glyoxysomes. Very commonly questioned in MCQs. Glyoxysomes. What are these glyoxysomes? They are specialized peroxisomes found in fat storing tissues. Okay, only in fat storing tissues of plants. Only in fat storing tissues of plants. Okay, then what do? You can write down, they convert fatty acids into sugar. So imagine you are maybe a certain type of a oil that is being stored in a plant. So you can have certain vegetable oils that are being stored in plants. Vegetable oils, canola oils, this sort of sunflower oils are found in plant tissues. So those are produced not for us to eat. අපිට මේ අපේ omega 3 fatty acid requirement එක fulfill කරගන්න. Not for that. Plants are producing those so that they can be used as energy storage when they need it. Right? So when they need energy storage, what they are going to do is they are going to break down their fatty acids to sugar using glyoxysomes. Right? Using glyoxysome enzyme, they are going to break down. Hari, can you complete whatever the page, this page, all the content about peroxisomes and glyoxysomes? Peroxisomes break down hydrogen peroxide into oxygen and water. Okay, කට්ටිය not sleepy නේද? Okay, we'll finish soon, huh? We'll finish soon. Let's continue. Mitochondria. Hari. So mitochondria and chloroplast is going to be an essay topic. Okay, mitochondria and chloroplast is going to be an essay topic. I'll be sending that essay marking scheme right now to the group. Okay, so I'll be sending it to the, not to the group, I'm sending it to the Zoom chat as well. So yeah, please download it. This is mitochondria and chloroplast essay. So everyone has got an essay writing book, neither everyone has got an essay writing book. So don't worry, it has it has two essays. Actually, the document has two essays. Therefore, you only need to go through the mitochondria and chloroplast essay right now. So you can see this is the document I'm talking about. Correct. So here it is about the first part is from meiosis, and so that you can do after we have done cell cycle. This is the essay that you have to write this week. Okay, so we are going to draw the diagrams right now, and you are going to write the sort of the text part of the essay at home. Right. So the first part is about chloroplast, second part is about mitochondria. So we are going to draw the diagram now, and I want you to write the essay topic. I mean, sort of the, let's say, the text of the essay at home, and you're at home already. Then you will have a nice complete essay. Correct. So what we are going to do is we'll draw the mitochondria and chloroplast right now. So before that, there's a small theory here. When we have a small theory, this is called the endosymbiotic theory. This is something you don't have in the syllabus, but when you understand this will be easier for you to understand some of the special features of mitochondria and chloroplast. Okay, if you take mitochondria and chloroplast, they look like bacterial cells. They have all the features of an independent bacterial cell, but the only thing is they are living inside your they are living inside another bigger cell. No, mitochondria and chloroplast they are actually organelles, but they look like an independent cell that is living inside another cell. So we think in evolution, at a certain point, there was a eukaryotic cell ancestor. Again, the modern eukaryotic cells that you have right now, this is the ancestor of that cell. Correct? So it didn't have a mitochondria or chloroplast. So it has nucleus, nuclear envelope, and all the other things. At a certain point in evolution, it engulfed oxygen using non-photosynthetic prokaryote, right? So you can see this is the engulfing. So later, so it was initially parasitic, right? It ate another prokaryote, but later that prokaryote started to live inside that eukaryotic cell. After many generations, became an organelle. The organelle now we call as mitochondria. Right? So that's how we think mitochondria originated. Then similarly, a photosynthetic prokaryotic cell could be another photosynthetic bacterial cell was eaten by this eukaryotic cell, and that became, with several generations, a chloroplast. Right? After several generations, it became a chloroplast. So that's how we think mitochondria and chloroplast originated to live inside your eukaryotic cells. Right? Okay, we'll come to the whiteboard and let's draw the chloroplast and mitochondria that you need to be able to draw. You need to be able to draw a mitochondria and chloroplast. It's a very important skill. Right? So mitochondria you have to draw while you are listening to me. Go through the resource book as well. While you are listening to me while I'm drawing this, please go through the resource book and read and highlight. Okay. So mitochondria, we have to draw it as an elongated organelle. Okay, elongated, like a Panadol pettak, right? It's a piece of like a tablet of paracetamol. Okay, so it's an elongated organelle like this. Right? Then it has two membranes. The outer membrane is the one I drew. It is smooth. The inner membrane has these foldings. It has become convoluted. Ralliatilla. And these convolutions are known as, can anyone tell me? What are these convolutions? They are called as cristae. Cristae of mitochondria. Okay, the function of cristae is to increase the surface area. Okay, then the space between an inner and outer membranes of mitochondria is called as intermembrane space. Very, very important. Intermembrane space. The space between the inner and outer membrane is known as intermembrane space. Maybe this inner and outer membrane, this space is called intermembrane space. That is it. It is very important in the next topic that we are starting in the theory class. We already started in the Saturday batch. That is cellular respiration. This intermembrane space is so important. Right? Okay, very, very important. Then, innermost part of the organelle is known as the mitochondrial matrix. So, similar to a cell cytoplasm, you have a sort of a cytoplasm inside the mitochondria that is called the matrix. So, matrix, this is the outer membrane, this is the inner membrane, is actually, this is the inner membrane, okay, and then what is this? Intermembrane space. Intermembrane space. Then you need to know what are the things included inside this mitochondrial matrix. In the mitochondrial matrix, you are going to have some DNA that we call as, which is very similar to the prokaryotic DNA. We call them as circular DNA. We call it as circular DNA, which is similar to the DNA you find inside a bacteria or a prokaryote. Okay. Then you have, because the mitochondria is mainly involved in production of energy, you can have phosphate granules that are needed to produce ATP, adenosine triphosphate. Then a lot of enzymes are there. Then also the matrix has a lot of enzymes that are needed for your Krebs cycle, or we call it as citric acid cycle, that we are going to learn very soon in the theory class, normal theory class. Okay. Cristae composed of proteins and enzymes essential for the electron transport chain and oxidative phosphorylation. Okay, so this is basically the structure of a mitochondria. You can label this as circular DNA. That is basically it. Yes, something big I missed. In this mitochondria, you are going to have ribosomes, free ribosomes. These blue color are the ones that I am drawing, are the free ribosomes. Can anyone tell me what type of ribosomes are these? These light blue are the ribosomes that I am drawing. What are the ribosomes that you have? What type? 70 is very good. Free 70s ribosomes are there. This is circular DNA. You can say these are phosphate granules maybe. No need to label actually. This is it. Okay, this is mitochondria that you need to be able to draw. Can you take a quick screenshot? I don't think I am going to give you some time when you are writing the essay. The essay topic you can write down. Online people are everyone. Okay, online. All new people, you need to have only one writing book for the theory class, for the bio class. That is a CR book to write essays. Okay, if you haven't written any essay previously, you can start with this one. The essay topic is describe the structure and function. Sorry, no need to function. Sorry, describe the structure of chloroplast and mitochondria. Describe the structure of chloroplast and mitochondria. That is the essay topic. You can start your essay writing journey with this. Others I think you have written. No? Okay, right. Shall we draw the chloroplast? Do we have to buy Pesuru books? No, don't need classified MCQs. No need classified MCQs, and also you only need to be able to, you can only do MCQs from 2019 onwards too. So Pesuru books are going to have from 2000 onwards or something. I think not even before that. Can you do those MCQs? No. Don't waste money on Pesuru for bio. Buy a good, invest on a nice print of the resource book. That's all. Campbell Campbell, I will send you a PDF if you want. Campbell, no need at all. Right? No need to buy definitely if you really want to read it. But I have included almost all the diagrams that you need from the Campbell in the tube. If you feel like you really need to go through it, I will send a PDF. I'm saying, okay, if you want to buy, you can buy. My recommendation is you won't have time to go through all this later. So your future self will laugh at you, right? What was I doing in these early stages, right? So you don't need to do Pesuru for bio because you're only going to be able to do, so including 26 paper and the 27 paper, a maximum of 8 past papers. The 8 past papers are going to be from your new syllabus, this new resource book. So those are the only questions you are able to do, and they are also the only questions you need to do. Nothing more. Bio is mainly essay writing practice, then go to the exam with the entire resource book in your mind. Guaranteed day pass. Sorry. Okay. We'll draw chloroplast quickly. Okay, read with me the structure of the chloroplast. Page number 33. So how you describe the chloroplast is like this. You describe it as a biconvex lens shaped organ. So I'll try to use mainly a bit of green color. Chloroplast is a biconvex lens shaped organ. Mehem. What do you mean by biconvex? It's like a lens, right? With the two outer sort of protruding layers facing outwards. So it has two membranes. Both membranes are smooth, unlike the mitochondria. The inner membrane is convoluted, but in the chloroplast, both membranes are very smooth. Okay. Then this is the outer membrane. This is the inner membrane. Then similar to mitochondria, you have a intermembrane space as well. Very narrow intermembranes. Very narrow intermembrane space. Inner membrane, outer membrane. And a very narrow intermembrane space. Right? Then inside the chloroplast, there's another membrane system. Inside the chloroplast, there is another membrane system. I think we talked about this a lot since we are doing photosynthesis. There is another inner membrane system that is inside the chloroplast. Now you can understand the proper structure of chloroplast. You guys have been joining the normal theory class, right? I need to make sure, okay, this Monday class is not going to continue. I think some people are still thinking this Monday up to speed sessions are not going to continue. They are not. Okay, you have theory classes on Saturday and Thursday. Please join with those. Okay, so this is the inner membrane system that is composed of a membrane called thylakoid. So this thylakoid is going to form flattened and interconnected sacs, one after the other. How I describe them is like rice bags stacked on top of each other. Rice bags stacked on top of each other. That is composed of thylakoids. What do you call them? These stacks are called grana. These stacks are called grana. Okay, then these grana are interconnected by another membrane, another membrane that is called as intergranal lamellae. These grana are interconnected by another membrane system, small membrane that is called as intergranal lamellae. Intergranal lamellae. Then we talked about how these for thylakoids contain these small enzymes as well as molecular units that are known as photosystems. We talked about those. Photosystems contain photosynthetic pigments and they are designed to absorb visible light. Okay, different wavelengths of visible light. Then, as usual, to a mitochondria, you have this circular DNA inside the chloroplast as well. Then you have 70S ribosomes. Again, you have 70S ribosomes inside this chloroplast. So, this chloroplast is similar to a photosynthetic bacteria, right? Photosynthetic prokaryote kenek wage that is living inside a plant cell, right? So, you can call this as 70S ribosomes. Yeah, what else? Then there could be some starch granules because why in the chloroplast you make food by the Calvin cycle, right? By the Calvin cycle. If you didn't join with our dark reaction photosynthesis, all those recordings are available in the July month website. Okay, July month course. Please go through them. Okay. So these you can just label them as starch granules. 70s ribosomes. There could be a bit of lipid droplets. And yes, then I forgot to label this in a sort of gel-like thing that is similar to the cytoplasm of a cell. In the chloroplast, it is known as stroma. Stroma. This is a nice chloroplast. This you should be able to draw and label and describe. Okay. Yeah, and you can put a label to one of these membranes and you can call it as a thylakoid. Anyone tell me if I am going too fast or anything you can't understand? Anything you can't understand? Quickly draw this or start to draw the sketch. Okay. So today I'm going to finish talking about cytoskeleton and then conclude for the day. Okay. So we'll continue there from next week, and then we'll next week we'll start cell division as well, mitosis, then meiosis, and then final day will be mostly from enzymes. Okay. Okay. Right. Then shall we come back to the our diagram dude? Anyone? All good, right? Any questions that you want to ask? You can ask. So we'll finish cytoskeleton and we'll conclude for the day. Okay. Right. Come to the resource book, page number 34. Let's read. Okay. Let's read. Cytoskeleton is the supporting structure of the cell and maintains its shape. Hurry. Highlight. It's the supporting structure of the cell and maintains its shape. It is more important for animal cells which lack cell walls. As you know, an animal cell just have a plasma membrane. No? It has just a plasma membrane. So therefore, it is very important that they have an additional way to maintain its cell shape as well as to get some additional support. Okay. Cytoskeleton is made up of microtubules and protein filaments, mainly microtubules, which are these guys, microtubules, and protein, certain protein filaments compose the cytoskeleton. Okay, let's see. It is dynamic and has the ability to break and reform as needed. Very important. The cytoskeleton is not something that is very rigid, right? So as the animal cell, or it could be a certain sometimes found in amoeba, certain protists like amoeba. So you know the animal cell should be able to be more fluid. Like if you take a plant cell, the plant cell is like a polygonal box එකක් වගේ, it is just inside the cell wall. Right? But an animal cell and sometimes the protist cells like maybe amoeba, they should be able to change its shape as well. At the same time, they should get some support. So that all these functions are done by the cytoskeleton because it is able to break and reform. It is not a very, it's not a permanently recomposed structure. It can break and it can regenerate as well. Got it? There are three types of components in the cytoskeleton as follows, as microtubules, actin filaments or microfilaments, and then intermediate filaments. So, actually in your page number 35, we in the previous edition of the same resource book, we had a bit more information, but it has been removed. So now you guys are, that is actually good because that is less information for you to memorize. Ne? So that's a good thing. The more, the lesser, the better. Okay. Okay, so we are going to do the comparison between the three parts of the microtubule, the cytoskeleton as well. Right. So, microtubules, okay, right, so this is what we call as microtubules. So, you can write down on top of it, on this box, you can write down as microtubules. You can see it is a tube, no? It's like a, you can say, what is it? It's like a pipe, like a PVC pipe. It is hollow inside, okay? It is hollow inside. And if you zoom in into the molecular structure, you can see it is composed of circulate, spiral shaped proteins, right? Those proteins have a name, they are called as tubulin proteins, okay? So the protein subunit, we'll write it in a different color. Protein subunit in microtubules is what? Tubulin. Easy to remember, no? Microtubules and tubulin. Microtubules and tubulin. Okay. Then you can say as the next point, they are hollow tubes. Okay. So you can write down they are hollow tubes. Wall is composed of 13 columns. Okay, you can see 1, 2, 3, 4, 5, 6, 7, 8. Okay, there's 13 columns of tubulin that are at a, they are spiraling around. So as they spiral along, as they continue to spiral along, you are going to get a nice tube like this. Okay, wall composed of 13 tubulin columns. Then their functions, okay. Then their functions are, these things we learn in cell division, okay. Form a type of, let's say, form the spindle. The spindle is something, is like the apparatus or the machinery during cell division which is going to move all the chromosomes into the daughter cells. The spindle is mainly composed of this microtubule. The spindle is like a machine or like a robotic arms that are going to move the chromosomes in the different directions when they have to go into the new daughter cells that are produced. Correct? So, form spindle in cell division. Also, next week we learn cilia and flagella. So, you can see in the microscopic structure of the cilia and flagella, all these are microtubules. Tubes. All these are microtubules. So, you can write here also form cilia and flagella and microtubules. Next component, you can call them as actin filaments or you can call them as microfilaments. You can put the topic here, actin filaments or microfilaments. So, you can see they are composed of a protein called actin. You know actin, when we did proteins, we talked about contractile proteins, we talked about actin, because actin is also found in your muscles. Okay. So, you can see you get a columns of actin that are wrapping around each other to form an actin filament. Okay. So, you can say protein subunit in actin. Filament is what? Actin. Okay. Easy enough to remember. Then you can say two intertwined strands of actin. Two intertwined strands of actin. Then what are the functions? You can write down what are the functions. There's a phenomenon called cleavage furrow. I'll show you cleavage furrow in cleavage furrow in cytokinesis. Right. Cleavage furrow is something like this. Okay. So, cleavage furrow is found at the end of cell division where you have a process called cytokinesis. So, the cleavage furrow is how the big mother cell is going to divide into two smaller daughter cells. So, what's actin going to do is, you can see there's going to be a ring of actin. There's going to be a ring of actin right here. Okay. So, all these actin fibers are going to pull the plasma membrane inwards. It's going to pull the plasma membrane inwards. So, that plasma membrane is going to get closer and it's going to connect and then you're going to get two cells. Alright? That's what we call as cleavage furrow. This is what we call as the cleavage furrow. Okay. That is done by actin filaments. Okay. So, where were we? In actin, no? Formation of cleavage furrow in cytokinesis, right? CYTOKINESIS, cytokinesis. Okay. Final point, we'll be concluding with this for today. Okay. Finally, our final component, what are we talking about? I have always mentioned this. As you keep on learning something, at a certain point, you are going to forget. what am I learning about? Okay, so right, sometimes when I'm studying maybe a certain drug or something, I'm going to be studying, okay, these are the doses, these are the side effects, these are the diseases I can use this for, these are the bacteria that I can use this for. Then at the end I forget what is the name of the drug I am studying. Okay, okay. So we are studying about the cytoskeleton. What is the final components of the cytoskeleton? That is known as intermediate filaments. These are the strongest components in the cytoskeleton. They are actually like cables. Okay, can you see? It's like a very strong cable or a steel cable thin. Okay, so it could be composed of several. There are several options for the composition. So you can say protein subunit. It can be one of several. A common example is keratin. A common example is keratin. Okay. Can you see? Keratins are tightly coiled together to form an intermediate filament. Okay. Then you can write down some features. It's a fibrous protein. Fibrous proteins. Supercoiled to cables. Supercoiled cables. Then what are the functions? Next week, first thing we learn about certain cell junctions, right? There are certain cell junctions that we call as a desmosome or an anchor junction. Here, this is that cell junction that we are going to learn about. You can see here, all these are your intermediate filaments. So this desmosome or anchor junction is a place where you need a lot of strength. So in order to provide that strength, you need the most strong fiber, okay, or the filament. So that's why we use the intermediate filaments in that desmosome. Okay, you can write here desmosome slash anchor junction formation. That's where you have intermediate filaments. So quickly take this down. Under our cytoskeleton is exactly like our skeleton. That is, they are to provide support to our body, to provide support to the body of the cell. You have this cytoskeleton. Okay. Any questions? Okay. Right. Okay, people. Then that is all for today. I think that's all we have time to cover today, right? So I was planning to actually go a bit further, but let's see. Okay, so tomorrow, next week is going to be up to speed session, almost 5 days now. Okay, so I'm hoping you are taking the maximum benefit out of these sessions, and let your friends know that they have, I mean, they can cover everything that we have done up to enzymes within just within 4 videos. They can cover everything. So you can let your friends know that there's such a program, and at the same time you can join in the theory class and balance everything out. Okay, so as I always tell you, this is the time to not to study hard, but to build your mindset. Okay, so to understand how the A level works, understand how the paper is composed, and understand how you have to work from this point on. Don't need to get stressed. Just study everything. Study means padang way. Just study how classes are done, go to several classes and see how it's been taught in several classes, and then sort of gather the information and then choose what's best for you. Okay, that's all I have to keep on telling you. But because in another year's time you will not have the time to go look for classes or to adapt yourself. Now you have the time to go for, to find out what is the best pathway that I can follow to go to the result that I want. At the end, in this exam, what matters is how much, I mean, how well you can write the paper. So as I always say, there are two ends to most of the classes. In certain classes, as they are just reading the resource book and nothing explanation, nothing is done, just going ahead. And in other classes, right, they are spending a lot of time on sort of just staying on small theory points, wasting time. Okay, so you need to have a proper balance between those. And as you can see, when you are joining our regular theory classes, I always try to maintain a balance between understanding as well as getting the topic done as soon as possible as well. Okay, so see you guys next Monday then. Any questions that you want to ask? Okay, then good night.